Excellent. All right. So again, welcome back everyone from spring break. We got a lot to do today. I was just getting ready to talk to you about the exams. Again, we're gonna have both the lab and lecture exam on Wednesday. And that is April 15th. And again, the tests are only available from 7 a.m. to 2 p.m. I still have to fine tune the exams. I have to basically really just check them to make sure there aren't any issues and problems along those lines. But for the most part, they are completed. So I wanted to give you an idea of what to expect from them so that you can plan accordingly. Your lab exam is going to have a total of 60 questions. Remember, just like on the practice exam, they are going to be presented one at a time. And once you answer a question, you cannot go back. It is going to be split evenly between the two systems. So 30 questions on the respiratory system and 30 questions on the urinary system. And they are both from large test banks. So I think I wrote close to 200 lab exam questions for this so that we would have the opportunity to be able to no, you cannot skip a question and go back on the lab exam. So you do have to, you are going to have to make a decision on a question and move on from it. They're going to be randomly selected from test banks. But again, there's no way for me to intermix the questions, so that's an advantage to you. So the first 30 questions are going to be on the respiratory system, and the second 30 questions are going to be on the urinary system. Now, it's just with these, you know, high, again, I tried to minimize the size of the images, but these are large images that you're going to be looking at. And so my concern is the timing of that to be able to go back and forth between questions. It'd be too hard to go back 10 questions and then to kind of come forward again. So one at a time, you cannot go back. So you must answer the question and move forward. And you will have a total of 70 minutes to complete it. So you can spend as little or as much time on each individual question as you want, but you'll have a 70 minutes total to be able to complete those questions. So you have to pace yourself. If you realize that you're on question 10 and you know you've already spent 40 minutes on the first 10 questions, then you're definitely gonna need to pick up the pace for it. Because at the end of the time, unfortunately, when we're doing the proctored exams, it does kick you out. So it's not possible to go over. There is a timer that shows so that you can see how much time. If it freaks you out, you are able to turn it off. But make sure you are paying attention to it. You cannot have your phone out, even if you're just using it as a timer. Make sure you use the timer that is on your exam for that. The lecture exam, on the other hand, will be a total of 30 questions. For those 30 questions, there are going to be 20 multiple choice questions. There are going to be 2 fill in the blank, and then there are 8 essay questions. Covering, again, both systems pretty evenly. With the lecture exam, all the questions are available. Let's say it this way, the whole exam is available at once. So basically, you will have all 30 questions in front of you, and you'll scroll up and scroll down. And so if you want to read the essay questions first so that you think about them while you fill out the multiple choice or whatever, if you want to skip one and go back to it, there's really no skipping. You just scroll up and down. All of the questions will be there. So that is, again, so these are both in this way more like what they are with our in-classroom exams. The lab exams are timed exams where you get one question at a time. Really, the only difference is there's no opportunity to go back for a review at the end of them. But the lecture exam, you have the whole lecture in front of you to be able to work with it as well. And for the lecture exam, you'll have 2.5 hours or 150 minutes to be able to complete that. What that means is that this exam, both of these exams together, the total time for that is close to 4 hours. So you want to make sure you plan accordingly. Now, honestly, this should be far more time than you need, but I would try to encourage you to set aside at least 3 hours to be able to complete both of these successfully. So I would encourage you to strongly start your exam somewhere between 7 and 11. Again, also you can take a break if you want to take your lab exam first or the lecture exam first, or whichever you want, and take that earlier and then take an hour break and come back and do the other one. That is fine, however you want to do that. If because the lecture exam's longer, you want to take it first to make sure you get it in. However you want to do this, you have that opportunity to be able to do that. So I want to encourage you to plan your day accordingly so that you can get those things in. Do not wait till 1:55 to start the lab exam, because again, the test is only available till 2. So that means you'll only have 5 minutes, and at 2 o'clock, you will get kicked out. So you must plan accordingly for that. All right. The last thing that I want to remind people about, again, is you cannot have any books, you cannot have your phone, you cannot have any other resources out. Also, when you are scanning your room, what I care about is the workspace that is around you. I don't think literally nobody showed their workspace around you. I have plenty of pictures, views of the corners of people's rooms, or their plants over on the side, or their pets, or some people roamed around the house with theirs. That is not what I want. You need to show me your workspace, that it is clear that you do not have notepads or electronic devices or other things like that. And obviously there should be nobody around helping you as well that way. So make sure that that is what you are showing me when you're doing those scans. So make sure to scan your work area. Other than that, like I said, it's going to pretty much work the way that the practice one did, only with less stupid questions than the practice one. Again, that was just an opportunity to see what the protocols would be and how it would be set up. All righty, so that is the game plan for Wednesday. We will not have lecture and lab, just like we normally don't on those days. However, that does mean when we come back to the digestive system, and we are going to have 2 weeks to complete it. So likely I will assign the pre-labs like I usually do for the digestive system so that we can hit the ground running when we come back the following Monday with the digestive system. And I'll make those materials available for you shortly after the exam as well. All right, I will probably get the exam posted. My guess is either tonight or sometime tomorrow. But again, it will not be available until 7 a.m. And again, it would just show you and tell you the information that I've told you, remind you about how it's going to present the information, the number of questions, and all of that. All right, questions on any of that? All right, excellent. Then let's go ahead and...
I actually had a question.
Oh, yeah.
Is this still worth 100 points?
Yes. Oh, great question. Yes. So the lab exam, because it is 60 questions, is going to be 60 points. And the lecture exam is going to be 100 points. Yes, that is. So it is going to be 100 points like our in-class exams have been, because the expectation is still that there will be some benefit to the final exam being able to help you with the previous exams. So yes, so I was able to get it to work out where it is going to be 100 points.
All right, thank you.
Excellent. Great question. Any others? Are the essay questions going to require us to draw graphs? No, as much as I would love to make you draw the oxygen hemoglobin dissociation curve, there isn't, as far as I've been able to determine, a way to do that. So you may be describing graphs or describing curves or things along those lines, respiratory pressures, how they change, those types of things. You won't be doing any drawing that way, but you definitely will be describing. So make sure, and again, this is one of the things I'm always talking to you guys about, make sure you're being descriptive, you're being, and this is going to be even more important on those types of questions. I expect I will still be curving it. It is more challenging in the way that it sets up because I'm not able to, I can't curve, I cannot curve the exam in the Proctorio, well, it's not really even Proctorio, in Canvas. In Canvas, there isn't an easy way to curve the grades, but I may do something to add points. Maybe we'll add a separate column where I'm able to add additional points into grades or something like that to compensate for how it would normally be curved. But I don't know. I haven't figured out. That's one of the things that several of the instructors and I have been discussing. We haven't found a good way to do that yet. So the plan is to still try to find some way to assist in that manner, but not sure what that's going to be yet. So like all exams, try to maximize your points on it. No, you cannot use a piece of paper for a scratch paper or anything like that if you're trying to do calculations. The calculation should be simple enough. You can either do it in your head or write it out in the essay question. One of the nice things about the essay question form is you have a big chunk of space where you can write it. So if you need to write it out there and to be able to do it, any math you would have to do should be simple enough where you should be able to do it in your head to be able to do that. There is, and I do for the lecture exam, there is a way to make a little whiteboard scratch paper come up, and I have selected that as an option, but when I've been previewing the exam, I haven't been able to do that yet. It doesn't work for me, and it may be because I'm doing it in the preview view, and when you're doing it in the preview view, you don't get the Proctorio thing. So there may be a little whiteboard, scratch board that you can bring up during the exam, but I have not seen that yet. But any math you'd have to do should be simple enough where you can do it in the essay space. And more importantly, if you're doing those kind of things, you need to show your work anyway, right? Because that's like how you get your points. I don't want just that, the expiratory reserve volume is, 3 liters. I want to show you how you came up with that number. So you're going to be writing it out anyway because you're going to be showing your work. So you'll just do it in that space. Awesome. Any other good questions? Or questions, period? Alrighty. All right, if that is the case, then let us switch gears to the lecture then. So let's go from sharing this to sharing that. We left off last time and we had started our mystical magical journey along the nephron. We had gone through all of filtration and talked about everything that occurs in the renal corpuscle, and we are on our way to the proximal convoluted tubule. And I need that. Thank you. That up there. Perfect. All right. As we were talking about, there are basically 5 major processes that occur in the proximal convoluted tubule. Again, the first of this is the reabsorption of organic nutrients. All right, we're going to talk a little bit about the pathways and the different ways that things can get back in through our proximal convoluted tubule. And obviously one of the major goals is the reabsorption of all the organic nutrients, all of the amino acids, all of the glucose, right? All of the small fatty acids that have come out, all those tiny little things that have come out that we need. We want to be able to bring those resources back into the blood because that is things that we want to hold on to. Again, in the last class, we talked about our transport mechanisms, both active and passive. And again, for those of you who didn't remember or were struggling with those, hopefully you've had the opportunity to go back and look at some of those. And they use primarily both active and passive transport mechanisms. Our active, like co-transporters, but then we also have our facilitated diffusion for those as well. We also need to have the active reabsorption of ions, right? Sodium, chloride, phosphates, things along those lines. Remember, as we talked about, it's not quite as important for calcium and also not quite as important for calcium as well. Remember, with calcium, we want that very strict value as well. So it's really important to make sure we have an appropriate amount of that. So it isn't about getting as much calcium back as possible as having the appropriate amount of calcium there as possible. And again, when we come to this, this is kind of what we were talking about in the endocrine system. If you remember when we were talking about the adrenal gland produces those glucocorticoids, it produces those mineralocorticoids, those types of hormones are some of the things that are going to help to drive that reabsorption of ions. But also it's going to be the levels of the ions in the blood as well. If we have massive amounts, then we're going to be releasing more of them into the urine, and if we don't have enough, then it's going to be transported back. And again, this is going to be a huge emphasis of that transport maximum we talked about last time. Remember, the transport maximum is how much of a solute we can bring back. If we have a massive amount of sodium in our blood, then what's going to happen is we're going to be transporting a lot of it back, but if it's above the transport maximum, then some of it is going to end up in our urine as a result of that as well. So again, that's how the ion levels in the blood determine, and that transport maximum determine how much comes back. We've got to clear those. Another major function of the proximal convoluted tubule is the reabsorption of water. Now, it does this using a special protein that can be expressed on the surface of the cells called aquaporins. Again, this is normally when I'd be drawing stuff on the board, but if we have, oops, I need black. If we have our cell, and of course, this is where we have our microvilli, so this would be the apical surface of it. What we are able to do is our cells of the proximal convoluted tubule have a special protein that they express on the surface that are known as aquaporins. And aquaporins are basically what allow water to be reabsorbed. Right? If you think about it, we have all of the stuff, the glucose, the sodium, all of those things that we were talking about, all of those things going back in. As they're going back in, then as we know, if all that stuff is being reabsorbed, water is going to follow. And water doesn't really have a choice in this either. The water has to follow. Let's do it that way. I like that. The water has to follow that stuff. In fact, we call this our obligatory, oops, now I messed that all up. That's all right. I'm just going to go ahead and erase it anyway. But we get the idea. What we call it is obligatory water reabsorption. Water is obliged to do it. It doesn't have a choice in this matter, right? I don't have a choice to stay home right now. I'm obliged to stay home so that, you know, grandparents don't die and I don't die and everybody else doesn't die. We are obliged to stay home. Water has to follow the stuff, right? That's something that an important concept we've been talking about since the beginning of 430. And that is this obligatory water reabsorption that occurs in our proximal convoluted tubule. And that's where we reabsorb as much as 65% of the water of our filtrate, which again, when you remember, we're thinking about making 200 liters of filtrate during the course of the day, reabsorbing 65% of that water is a significant amount. This also plays an important role in increasing the remaining concentration of the solute left behind. If water is leaving the filtrate, then that is going to concentrate the remaining ions that stay behind, and that sets up a concentration gradient. And that concentration gradient is going to help to allow for some of the passive reabsorption of ions. We are going to get, and this is one of the ways, for instance, that potassium gets its way back in, or some other things are able to get their way back in. They're able either to passively come through leak channels, or they're able to sneak between the cells. Remember, we talked about how there are those tight junctions that limit the movement of solutes, but doesn't completely restrict it. And so there are some materials that can be reabsorbed passively between the cells via that paracellular passive reabsorption set up by that concentration gradient. And then, of course, lastly, as we talked about, our proximal convoluted tubule is both where tubular reabsorption and tubular secretion takes place. The majority of them both take place in the proximal convoluted tubule. So when we talked about the three processes of blood filtration, glomerular filtration, tubular reabsorption, and tubular secretion, we numbered them 1, 2, and 3. Remember, they're really 1, 2A, and 2B, because secretion and reabsorption are occurring at the same time. And remember, the majority of both occurs in the proximal convoluted tubule. Now, it's great to talk about these things in general, but we also want to give some specific examples. In fact, I guarantee one of these specific examples is going to be an essay question on your exam. So let's talk about some of the ways that some of these, oh, thank you very much. Some of these materials are going to be reabsorbed. And let's talk first with sodium. Sodium by far is one of the most important. Ions that is going to be reabsorbed. As we know, there is a large amount of it in our extracellular fluid. And as we talked about in muscles, as we talked about in nerves, sodium entering into the cell plays a huge role in producing action potentials, things along those lines. So it is super important for that. But the other reason in particular that it's important in the urinary system is because, again, as we talked about, there is a huge driving force on sodium. Sodium really, really, really wants to get into the cell. And we can use that driving force of sodium to do work. So not only is sodium vital for the processing of a lot of, or for a lot of physiological processes in the body, and we need a lot of it in our extracellular fluid, but in this case, that huge driving force, we're going to be able to use work. Now, typically, sodium is brought in. And again, remember, one of the important things when we're talking about all of these transports, and we mentioned it in the last class, but we kind of split this one in half. We are going to use different mechanisms on both the apical and the basal surface of the cell. So again, I'll do let's go back to, here, let's cheat real fast and go back to our whiteboard. Clear all of that. And do a quick drawing to remind ourselves of this. So we have our cell. And we'll put some microvilli on this so that we know that this is going to be our apical surface. That's way too big. There we go. So this is our apical surface here, and this is our basal surface. And remember what that means. That means out here is our filtrate, right? Or let's say it this way, is the lumen of the proximal convoluted tubule. And it contains our filtrate. And then in here, and I gotta move this stuff out of my way. We have our paratubular capillary. Let's try that again. Peritubular capillary. That's here. And then, of course, this is our interstitial fluid. Excellent. All right. Our goal is to basically bring things in. And again, the way we breathe things in across the apical and across the basal surface is going to be different. So with sodium, we are going to primarily use pumps. Some of those pumps we'll just directly use, so we can have some primary active transport where we are using ATP to bring in that sodium. So that is one thing that we can do. But the other thing that we can do, as we've talked about, is we can use secondary active transporters. Secondary active transporters also allows sodium to come in. But when we bring that sodium in, we use that to do some type of work. And I will use a double-sided arrow to emphasize that it is either going to bring something in or bring something out. Again, it's not going to do both. So we'll remind us that with a big fat or here. It's going to be one or the other. But we use the energy of that high driving force of sodium wanting to come inside to do some work for us. All right, so this is on the apical surface, this is what we're doing with sodium. So let's go back to lecture and see that. Move this up here. I need my annotation tools back. Perfect. All righty. Once it gets into the cell, we need to kick it out. Because as we know, we want a large amount of it in our interstitial fluid, low levels of it inside. So here, while we can, even if we're using that co-transporter to bring sodium in, we're not directly using ATP. We are going to directly use ATP to kick that sodium out. And of course, when we kick the sodium out, potassium comes back into the cell. But remember, one of the things we talked about is potassium isn't as important. So one of the things that happens is that potassium is able to leave directly via leak channels again. So what this does is basically just circles the potassium around. It doesn't directly affect the concentration of potassium inside the cell or inside the interstitial fluid. But what it does do is constantly drive sodium out of the cell. And if it constantly drives sodium out of the cell, we have a very low level inside of the cell. And that, like I said, is going to be our huge driving force that we're going to be able to use to do work. All right, so again, not only is it important to bring back sodium because we want that large amount of sodium in our blood, we want that large amount of sodium in our interstitial fluid. But this process also sets up this huge gradient, which is going to allow sodium to do work to bring other things back in for us. All right, so again, keeps our intercellular sodium very low, produces that strong gradient that we can use for our co-transporters. All right, questions on that? All right, let's talk about one of those transporters. All right. Now that we know how sodium gets back in, we can talk about and see some of the other pathways of how things get back in as well. And again, as I mentioned, one of these pathways is going to be an essay question. So either the sodium one we just talked about or one of these right here. Another common pathway is glucose, and really the way glucose comes in is how all of our organic nutrients come back in as well. So that means things like amino acids or other types of things. Oops, I need to erase that. There we go. So again, other organic solutes come back in this way as well. And if you notice with this, Oops, we'll get to that in a second. All right, so let's actually see and talk about the pathway. Notice it comes in on the apical surface with a co-transporter. Oops, is it up so I spell it right? Notice this co-transporter is letting sodium come in. So we talked about sodium really wants to come into the cell. Once it's in the cell, it is then kicked out by our sodium potassium ATPase, and that produces that huge concentration gradient. So, all right, sodium, you really want to come into the cell, but you're going to do some work for us. You're going to bring sodium in. Remember, as we talked about, this is an active process. It's a secondary active process because the ATP is used down here on the basal surface. But the key is this still uses energy. So if you think about it, we are using energy on the apical surface. And this is important because remember, any glucose that stays out here in the filtrate is going to leave the body as urine. So it is important to use energy to cross this apical membrane, because once we cross this apical membrane, then our glucose and our other organic nutrients have been saved, right? Once they're inside the cell, they're safe and they're not going to be lost as urine anymore. So it's worthwhile to use that energy to expend that resource to save that glucose on the apical surface. However, once we've saved that organic nutrient, that glucose, that amino acid, once it's inside the cell and it isn't a risk of, there isn't a risk of it being lost, we don't have to worry so much on the basal surface, right? The more we save, the higher amount, the larger the concentration we're going to have inside the cell. And with that larger concentration we have inside of the cell, that is going to produce a diffusion gradient, and that concentration gradient is going to allow it to just passively leave the basal surface via facilitated diffusion. And then, of course, once it's in the interstitial fluid, it enters into the capillary. And it is saved. So notice, remember, as we talked about, we are using energy to reabsorb these materials, but only on the apical surface. All right, questions on that? All right, then let's talk about the fun one. The fun one is hydrogen secretion. Although hydrogen secretion is really not so much about secreting hydrogen, right? We're talking about reabsorption here. What we're really talking about is the reabsorption of bicarbonate. Now, as we look at the picture here, it can be a little intimidating. So let's go back to my drawing and steal from this. So let's use this drawing to talk about the process of the reabsorption. So I need to, let's see, get rid of that. Oh no, didn't want to get rid of that. Undo. There we go. Oops. All right, now I don't know what happened. Undo. There we go. Okay. So I want to erase that. I want to erase that, and 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 that. Excellent. All righty. So out here in our filtrate, we have bicarbonate. And that bicarbonate is something we want to reabsorb. Turns out the way we do that is by kicking hydrogen out of the cell. So again, we have one of these co-transporters. And in this case, it's an antiporter. What it does is it allows sodium to come in, because sodium really wants to come into the cell, and we use that sodium coming into the cell to do work for us. And that work that it's going to do for us is, in this case, going to kick a hydrogen out. So we get rid of that hydrogen ion. All right. Again, one of the things we want to do is we want to maintain the pH of our blood. And if we're going to maintain the pH of the blood, we obviously can't make it too acidic. So getting rid of that hydrogen ion is definitely a good thing. However, as we just learned in our respiratory system, right, when these kooky kids, hydrogen ions and bicarbonate, come together, they form carbonic acid. And as we know, carbonic acid is able to dissociate into carbon dioxide and water. Now remember, as we learned in the respiratory system, this is a two-way chemical reaction. It can go in either direction. However, in this case, what is happening is that with hydrogen ions being kicked out here into the lumen, we have an increase in the amount of hydrogen that is coming out here. It is going to mix with the hydrochloric acid. And as a result of that, we are going to drive the chemical reaction in this direction. And as a result of this, so, oops, don't want to do that. There we go. Just wanted to lift this up a little bit. There we go. The result of this process is that basically what we end up doing is we produce CO2 in the filtrate, right, out here in the lumen. So the result of this process of kicking out that hydrogen is we make CO2 out here in the lumen. All right, you guys with me so far? If you're lost now, it's just going to get worse. So any questions? Yes.
What is the increase? So like, is it an increase in the net concentration of hydrogen ions? Okay. And
absolutely.
So that's,
yeah,
okay.
when we're kicking these hydrogen ions out, that basically means that there's now more hydrogen ions out here in the lumen, right? So we're getting an increase in concentration, and more hydrogen ions then can mix with our bicarbonate and drive this two-way chemical reaction to the right.
Okay.
Yeah, exactly. That's the whole point. The whole point of this pump of the sodium, is that by kicking hydrogen out, we increase the amount of hydrogen that's out here in the filtrate, and that drives the reaction in this direction. And so we produce CO2 as a result of this. So out here in the lumen, we start producing CO2. Great question. Any others? All right, excellent. So now we have this CO2 that is being produced. And there's obviously more of it here in the lumen than there is in the cell. And as we know, CO2, being a small nonpolar gas, is able to basically enter the cell, via simple diffusion. And it does it down a concentration gradient. All right, move that up here. So our carbon dioxide is going to enter into the cell because we're making more of it here, right? There's now more carbon dioxide out here inside of our lumen. So it moves down its concentration gradient, and that CO2 enters into the cell, right? So let's say it that way. There's now more CO2 inside the cell. All right, you with me so far? Excellent. Blinking spaces. I mean, we totally get this. Awesome. Well, guess what else is inside the cell? Water. And what happens when carbon dioxide comes in contact with water?
Carbonic acid.
We produce carbonic acid. Excellent. And that carbonic acid is then able to dissociate into hydrogen ions and bicarbonate. Again, this is a two-way chemical reaction, but because we've got more carbon dioxide coming into the cell, that drives the chemical reaction in this direction. And I want to actually move that up here. So now it drives in this direction, and we get hydrogen ions and bicarbonate. Of course, with those hydrogen ions are going to get pumped right back out again. So once we make a hydrogen ion, that hydrogen ion is going to get pumped out by that antiporter with sodium. And what that leaves us with is the bicarbonate. And so what we are going to do is then release the bicarbonate from the cell on the basal surface. And in this case, we are able to recapture our bicarbonate, which is an important blood buffer. Right? And in the process, we release hydrogen ions, and that maintains our blood pH. So in this process, not only are we recapturing our bicarbonate, where we are able to maintain that important blood buffer, but it also plays an important role in helping to maintain the pH of our blood. And now that I've done this, and I'll go ahead and save this whiteboard image, and I'll be posting these. No, great question. So, I'm sorry, I guess I didn't do that. Yeah, let's go back, okay. So, notice we don't want, so yes, we have hydrogen and bicarbonate. But remember, if we started to have an accumulation of bicarbonate inside of the cell, then when we have an increase in bicarbonate, that could start driving the chemical reaction back in the other direction. So in this case, we do not want to be passive on the basal surface. So instead, we are going to be active. And here we're going to use a co-transporter. In this case, that co-transporter is going to be a symporter. And what we are going to do, great question, thank you for asking that. What we are going to do is we are going to kick that bicarbonate out, and we're going to do it well, it's actually no, I'm sorry, it's not a co-transporter. Let me rephrase that. I gotta select this. It's one of those times we're trying to write and talk and draw and do all these things at the same time, screw things up. So let's erase that. That's the easiest way to do this. This is an active pump. And this active pump uses ATP, and it uses ATP to kick both sodium and bicarbonate out of the cell. So here, both bicarbonate and sodium are kicked out, but this is not a co-transport. This is actually a pump. This pump is using ATP. Because remember, sodium is not going to want to leave the cell. So we're going to have to use that ATP to kick sodium out, and the bicarbonate comes out as well. And so as a result of that, our HCO3 is now released into the interstitial fluid. And once it's released into the interstitial fluid, it's pumped back into the blood. Notice too, and thank you for asking that question, because that was the other point that I wanted to make. Notice with things like glucose and amino acids, while those things are definitely important and we use the active transport on the apical surface, once they're safe inside the cell, we can just let them diffuse out of the cell slowly and get recaptured that way. But not our bicarbonate. Bicarbonate is we use energy on both the apical and the basal surface to move it. So that removal of hydrogen, that maintenance of our pH of our blood, that getting back that important blood buffer is a very important process. Thank you for asking that. Great question. And there is, oh, and I guess I go, hold on. Before I forget, let's go back to the whiteboard and I'll take the picture with the updated. And these pictures I'll post on our Canvas site as well. So I have that. Perfect. And again, if you don't like my art, here we see the pretty picture from your textbook showing that exact same process. So again, we start with that antiporter, sodium with its high driving force coming into the cell, kicking the hydrogen out. That hydrogen ion mixes with the bicarbonate out here in the filtrate, drives our chemical reaction to make carbon dioxide and water. That carbon dioxide, being a small nonpolar gas, passes through the apical surface just using simple diffusion. However, once inside, it mixes with the water inside the cell to produce carbonic acid, to dissociate into hydrogen ions and bicarbonate. That hydrogen ion can then be pumped back out again, and that bicarbonate is going to be actively pumped out the basal surface along with sodium to be reabsorbed into the blood. So like I said, if you don't like my pretty picture, your book's got a pretty picture that shows this process as well. Alrighty, so like I said, one of those three processes, the reabsorption of bicarbonate, the reabsorption of sodium, and the reabsorption of basically organic solutes like amino acids and glucose will be an essay question on the exam. All right, questions on that? All right, excellent. And then here again is a pretty picture showing the summary of all of these things. Again, it shows also our paracellular reabsorption and things along those lines as well, talking about all of these processes in the proximal convoluted tubule. And then your book's got some nice tables that talks about these as well. All right. By the time we are done in our proximal convoluted tubule, our filtrate has now been dramatically changed as a result of this. All right. Because again, remember, most of the processing takes place in the proximal convoluted tubule. At this point, 100% of our glucose has been reabsorbed. At this point, 100% of our amino acids and other type of organic nutrients have been reabsorbed. About 65% of our sodium has been reabsorbed. And again, not surprisingly, if 65% of our sodium has been reabsorbed, 65% of our water has been reabsorbed as well. A close number with our chloride, but notice not nearly as much as our potassium. Again, our potassium isn't quite as important as the others are. So at this point, basically, we have a lot less filtrate with a lot less stuff. At this point, it's basically electrolytes, really salts, and water at this point, right? And then obviously any type of toxins and other things that need to be secreted as well, but we'll talk about those when we get to the distal convoluted tubule. All right, so it is this dramatically changed filtrate that is then going to enter into our loop of Henle. All right, Now, as I mentioned, we do need to talk a little bit about tubular secretion, but as we also talked about, it's essentially just reabsorption in reverse. It's using the same type of mechanisms, co-transporters, antiporters, facilitated diffusion, and active pumps, all of those things to move. And its job in this case is to get rid of substances that didn't come out in the glomerulus. Right? Remember, one of the things we talked about with that glomerular filtration is that it is a passive process. And that means we can't be selective. So basically, it was just all the small stuff, good or bad, went out. That means there's plenty of good small things we need to bring back. It also means there's plenty of big things that are bad that we need to get rid of. Some of these substances are large plasma proteins or substances, toxins, things like that, that are bound to plasma proteins, so large components that way. Another important component is the removal of our metabolic wastes. And these are urea and uric acid. Urea is a really interesting molecule. We could spend a whole lecture talking about urea. It was the first organic molecule that was created in a lab. Early on in the study of the body, one of the things that was thought is that one of the big differences between living organisms and non-living organisms is that living organisms had some inherent characteristic to them, some life force or something along those ways to them. So when people were able to actually produce a biological molecule in the lab, and when they were able to do that in urea, they were able to ultimately disprove that. So they were able to show that. It's an important metabolic waste. And again, these things are important because the same way that CO2 is a waste, but if you remember in our respiratory system, our goal wasn't to get rid of all of the CO2. Our goal was to use CO2 because it helps to maintain the pH to keep it under tight control, right? The partial pressure of CO2 fluctuates between 40 and 45. It's a very narrow range because it's so important for maintaining pH. The same thing is true with urea and uric acid. Urea and uric acid are metabolic wastes, but they also are a big part of the stuff that helps to maintain the osmolarity of the blood and the interstitial fluid. So one of the things that happens is it's small. So a large amount of it gets pushed out during glomerular filtration. However, in the proximal convoluted tubule, what ends up happening is all that active movement of sodium and glucose and other organic nutrients and things like that back into the body sets up that pressure, that concentration gradient that we talked about that allows small things like urea and uric acid to sneak back in through those paracellular cracks. So they're able to get back in. And so once they get back in, we then have to kick that excess back out again. So urea cycles dramatically within the urinary system. Like I said, we're not going to go nearly into as much depth as we could. We could spend a whole, not a whole semester, but a whole lecture talking about it. And in fact, while it plays an important role in what goes on in the loop of Henle, we're not going to talk about it there as well. Although we'll say your book does a nice job of talking about it. But so if you're interested in that, you can read that. So we need to remove substances that have snuck back in via those passive paracellular processes. We have to get rid of excess calcium and excess potassium. And then obviously, we play a major role in helping to regulate our blood pH. So and again, of course, also there is the removal of toxins. And by toxins, I mean anything that is not naturally occurring in the body. That can mean, drugs. That can mean medicines, right? All those types of things. Anything that basically is not biologically produced by your body. Right, it could be the pesticides that were on the lettuce that you didn't wash off properly before you ate it, or any of the kind of things along those lines that are going to be reabsorbed that way as well. All right, and that is going to bring us to the loop of Henle. The loop of Henle is going to take quite a bit of discussion to talk about. So I think even though it's a little bit early because we talked a little bit about the exam, I think this is a good point to take our first break. So what I'm going to do is go ahead, unless there's any questions, I'll answer any questions. But then if there's no questions, we'll go ahead and take our first break. So any questions? All right, so then let's go ahead and take our first break here. It looks like it's 9 o'clock, so we'll come back at 9:10. And at that point, I will restart the recording. All righty, see you guys. Go take a stretch, get a cup of coffee, splash some water on your face, and then I'll meet you back here in 10 minutes. All right, excellent. So let's go ahead and start talking now about the loop of Henle. Remember, as we've talked about, there's been a huge change in the composition of our filtrate by the time we've reached here. But one of the issues that we've had along the way is that, as we talked about, basically anywhere the solutes went, water went with it. So one of the big advantages that we have here in the loop of Henle is here in the loop of Henle, we're finally going to be able to decouple the movement of water and the movement of ions. So this is going to be really helpful for us to be able to help to maintain, as we said, one of the big concepts is not just about having the right amount of sodium and the right amount of potassium and all those things, but we also need the right amount of stuff. One of the big concepts that our loop of Henle is going to help us with is going to help us to maintain the osmosis, or really osmolarity, of the blood. But as we're going to also see, the other thing that the loop of Henle is going to help us to be able to do is to, excuse me, control the volume of the blood, right? And we see that, and again, it's a little tricky because when we talk about, a lot of times we talk about the loop of Henle, what we talk about is really the production of dilute or concentrated urine, right? But remember, the goal isn't necessarily to have a particular concentration or a particular diluteness to our urine. It's really about controlling the volume of the blood. If you are well hydrated and there's plenty of fluid in your body, then we need to get rid of that excess fluid. And so we produce a large volume of dilute urine, right? Or if you are underhydrated, then our goal is to try to recapture as much of that water as we possibly can. And that dramatically decreases the volume of the urine that is produced and therefore concentrates the urine as well. So this decoupling of the movement of water and ions is going to occur here in the loop of Henle, and that's really going to help us to be able to control things like the osmolarity and the volume of our blood. So let's go ahead and clear that, and then we can say these things over again. Again, like I said, 60% of the filtrate has been reabsorbed in the proximal convoluted tubule. So here in our loop of Henle, it's mostly the movement of ions, in particular sodium and chloride, and also trying to reabsorb the remaining water, but again, decoupling, pull them apart, teasing them apart from each other. It's able to do this by using what we call a countercurrent mechanism. What we mean by a countercurrent mechanism is, let's go to our whiteboard and go ahead and clear all of that. And we'll cheat and draw it sideways here. What we mean with a countercurrent mechanism is basically you have the flow of two solutions that are going in opposite directions. A classic example of these is something we call a heat sink. Basically what happens is we have, right, very hot water that passes through that tube, and what happens is we take then cold water and move it in the opposite direction over the top. what happens as these two things are in close proximity to each other, the cold water going by steals heat from the hot water that's going by. And so at the end of this process, we have warm water that is working its way out, or even cool water that's coming out the other end of it. And of course, this water gets warmed up. So now we have our warm water coming out the other end of that as well. And so that is what's going to be happening here. In this case here, we have these two solutions moving in opposite directions. And of course, in our loop of Henle, we have that thin descending limb and then that thick ascending limb. And so we have those two opposite movements where we have the water moving down one side, or filtrate moving down one side and then coming back up the other side, where they're going to be able to interact with each other. So it is this interaction of these two opposite moving flows that lead to what we call this countercurrent mechanism. And in this case, there's really two that are occurring here in the loop of Henle. The first of these is what we call the countercurrent multiplier. And basically what happens here in the thin descending and thick ascending limb, I keep losing that tool, there it is that in the descending limb, water is basically pulled out of the filtrate. And in the ascending limb, it is ions that is pulled out. So what is happening on the one side is different from what is happening on the other. So if we go back to our illustration, let's do that. I'm not sure if it's better to do this in the drawing, or let's just go through the pictures. We'll just walk through this, and then if it's confusing, we'll do it again. So we have our loop of Henle. And again, one side is thin, and the other side is thick. As we know, on the thin side, it is simple squamous. Cells. And on the, excuse me, let's go ahead and write it out here. Thin descending. And on the thick ascending, we have our simple cuboidal cells. And as we just mentioned, these simple squamous cells are impermeable to ions, but permeable to water. So what that means is water can come out, but ions can't. Oops, there we go. So water is able to come out of the thin descending side, but ions cannot. The simple cuboidal cells are the opposite. These are impermeable to water. In fact, basically, from the thick ascending limb all the way through the distal convoluted tubule, all the way to the collecting duct, our entire nephron is impermeable to water from this part on. However, it is, and I'm going to put in parentheses, permeable to our salts. And I put it in parentheses because really what we're doing here is we are going to actively pump the ions out. So on our thick ascending side, what we are going to get is the active movement of ions. Oh, wow, that was really bad. There you go. Active pumping of ions, and let's be more specific, sodium and chloride. that are going to be coming out of our thick ascending limb, but no water. So like I said, our goal here is going to be to decouple these two processes. All right. I think it makes a little more sense to talk about this rather than starting where it first enters from the proximal convoluted tubule, but to start down here at the bottom. Right? When we start down here at the bottom, we have a filtrate. And if you think about it, the filtrate down here, even though we haven't talked about exactly how that process has occurred, has had water being drawn out of it. So if you think about it, if you have a gallon of salt water, right, and you pull out half the water but leave all the salt behind, you're going to have a half gallon of solution that is going to be much higher concentration than it was before. Because what we have is the exact same amount of solutes, but less water. So our filtrate down here at the bottom has a high concentration of stuff, or again, like we talked about, it then is going to have a high osmolarity. So our filtrate down here at the bottom is going to have a high osmolarity. So as that filtrate is moving up the thick ascending limb, what we're going to do is we're going to use ATP to pump the sodium and the chloride and also the potassium out. However, what's going to end up happening is there are potassium leak channels, and those potassium leak channels allow potassium back in. So we don't really get a major movement of potassium here. What we get is a movement of sodium and chloride. And what happens is you have a bucket that has 100 sodiums in them. And let's just talk about sodium because it's simpler that way. And you've got a bunch of cells wanting to pump it out. So you pump a lot of sodium out. You had 100, and now you have 75 because you pumped 25 out. Up here, you're going to pump some out as well, but there's less left. So notice what's going to happen as we go up. There's going to be less and less sodium left to pump out. And so while there's going to be lots of sodium that comes out here, this is going to be a little less. And then this is going to be a little less. And then this, oops, what happened there? Little less. And so on and so forth as it works its way up. This is basically going to do two things. By the time our solute reaches the top, by the time our filtrate of our loop of Henle, it has a low osmolarity, right? Which is fancy way of saying it has very few ions in it. And we can actually give a value to it. That value is going to be about 100 milliosmoles per kilogram. Now, of course, that number in and of itself doesn't really tell us much unless we have something to compare it to. In our blood, in our interstitial fluid, and in our filtrate, up to this point in the proximal convoluted tubule, the osmolarity of our blood is about 300 milliosmoles. Again, it's that way in our blood and interstitial fluid, in all the cells in our body. That's the amount of stuff that we have in relation to water that we have in there. And again, because, let's put a big asterisk here, of the obligatory water reabsorption, anytime a solute left, water left with it. So what that means is that the osmolarity of our, oh, I didn't the osmolarity of our filtrate would not have changed. And that's why we need to decouple it here in our loop of Henle. So it starts as 300 entering in, right, then gets concentrated on the way down, and on the way up, it is going to be actively pumped out. And so we're going to get this change of gradient. So it does two things. The two things that it does, like we said, is it dilutes our urine. So we only have 100 milliosmoles of stuff in our urine, or in our filtrate, what's going to become our urine, on its way to that. But this unequal pumping of sodium does one other thing as well. It produces, let's use green for this, a osmolarity gradient in the medullary pyramid. Again, this is particularly important in our big juxtaglomerular, juxtamedullary nephrons, with those big, huge, massive loops of Henle that go deep into the medulla. This unequal pumping of sodium basically produces a concentration gradient, an osmolarity gradient in the medullary pyramid. In the cortex, like we talked about, it's 300 milliosmoles. At the top of it's 300 milliosmoles. But by the time we get to the bottom of the pyramid, deep in the pyramid, it can be as high as 1200 milliosmoles. It can be 4 times as concentrated down here at the depths as it is up at the top and in the cortex. So we produce this osmolarity gradient. Oh, I don't want that to be red. Oops, didn't want to do that either. There we go. We produce this osmolarity gradient. And again, I'll show it to you with all the pretty words and all the pretty pictures in just a minute. I know I'm making a mess of the board here. But we have this osmolarity gradient. And what this osmolarity gradient then is it drives the movement of water. If we come back to the thin limb, remember, as our filtrate is coming down inside of this, it is permeable to water. So if you think about what happens, if at this point right now, remember, our filtrate coming in as it entered was at 300 milliosmoles. And so if right here at this point, and let's go ahead and use a stamp. At this point right here, if it's 300, but outside, it's 400 milliosmoles. Then what's going to happen is, as we know, there's more stuff outside than there is inside. And so what's going to happen is water is going to come out. And that water is going to come out till it reaches equilibrium. And that equilibrium is going to be when it's at 400 milliosmoles inside. And then it goes down here, where there's 600 milliosmoles. And at this point here, again, water is going to come out, and it's going to continue to come out until it equals the 600 there, and so on and so forth. And remember, as we talked about at the beginning, when our filtrate is down here at the bottom of our loop of Henle, it has a high concentration of stuff, a high osmolarity, and it can be as high as 1200 milliosmoles. And then, of course, it is actively pumped on the way back up. Notice, because it is actively pumped on the way back up, we are no longer reliant on what is going on the outside. So out here, it may be, 500 milliosmoles out here. But because we're actively pumping things out, it may be 250 here on the inside. Because again, we are no longer reliant on the water forcing itself to move out on this side. We're actively pumping it. Water cannot follow. So we can pump things out more active. And that's how we're able to get a filtrate that is more dilute than the blood and the surrounding interstitial fluid. This is a mess. I'm going to go ahead and save this picture just in case it's worthwhile. But let's take a look at the pretty pictures from your book and see if that helps to make more sense of this. So let's do this whole thing again. I got to bring back all of you. And that. Perfect. Alrighty. So again, as we talked about, our thick segment of the ascending limb is simple cuboidal epithelial tissues. And here we are actively pumping sodium and chloride out of the filtrate and into the interstitial fluid. Now notice, as I mentioned, that active transporter actually moves chloride, sodium, and potassium. But notice, to get the sodium out, we use that sodium potassium ATPase that kicks sodium out and brings potassium in. And as I also mentioned, there are leak channels that can allow that potassium to go back out. So one of the important things to remember about this process is being driven by sodium and chloride. There is no net change in potassium levels from the loop of Henle. It is just sodium and potassium. So we keep talking about sodium and potassium. It is important to remember that this co-transporter, this is an active transporter, it is a pump, directly uses ATP, and it moves all three. But potassium can then leak back out. All right? And this thick ascending limb does not allow water to go through. There are no aquaportants. So in this case, when we move the salts out, water cannot leave. So our sodium and chloride are released into the interstitial fluid of the medullary pyramid. And as we talked about, that causes the osmolarity to be raised. It sets up an osmolarity gradient. Your book's got this great picture that does a really nice job. In fact, your book's got this great in focus. I was just looking at it this morning. Oh, got my water. It's focus figure 25.1, starts on page 996, does a really, really excellent job of talking about and showing this process. So if my explanation, the way we're doing this is confusing, I strongly encourage you to take a look at it in your textbook. Your textbook does an excellent job of describing this. But as we can see, this active pumping of sodium and chloride out of the thick ascending limb, got to clear all my drawings, sets up this osmolarity gradient in the pyramids. And again, we see that here when we take a lower magnification view, as we talked about out here in the cortex, and for that matter in our blood and our interstitial fluid, our osmolarity of our blood and our interstitial fluid is about 300 milliosmoles. However, in the pyramids, thanks to those juxtamedullary nephrons, and that thick ascending limb, we're able to produce this huge concentration gradient. And this huge concentration gradient is going to drive the reabsorption of our water. So actively pumping that sodium out leads to the active reabsorption, well, not active, but the osmotic reabsorption of water in this process. All right. So we've set up this concentration gradient. Excuse me. So now on the thin limb, when the water is coming down, pardon me, when the filtrate is coming down, as it gets to an environment where the osmolarity in its surrounding is higher than inside of the filtrate, because our thin descending limb is permeable to water, but not salts. Obviously, salts would want to come in because there's more of these salts outside here than there is here, so they'd want to come in, but they cannot. It's impermeable. So instead, what happens is water goes to the stuff. So water is drawn out. And as that water is being drawn out, right, water gets drawn out, that increases the osmolarity, that increases the concentration of the filtrate that stays behind. So water is drawn out by that osmolarity of the interstitial fluid, concentrating our filtrate, which then allows massive amounts of sodium to be pumped out down here at the deeper parts, and then less and less as it works its way up. And the process continues. So notice we have this countercurrent mechanism. What's happening in one influences what's happening in the other. Even though these two things don't touch, they're close enough to influence each other. But remember, I also said that there are two countercurrent mechanisms taking place. The second one, and we'll talk about it in just a minute, is what's going on in the vasa recta. Remember, when we talked about those juxtamedullary nephrons, we know they have this big, huge lattice-like or ladder-like capillary that is draped over the top of them, and that is the vasa recta. The vasa recta does two things. It brings the oxygen for all this active pumping of the solutes that is taking place here. But the other thing it's going to do is it's going to help to maintain this osmolarity gradient, right? Because if you think about it, if there is a, as water comes out into this, you're increasing the water to the solutes that are already there, you would think that it would be diluting this area and it would change, right? As we know, if you have a concentration gradient or an osmolarity gradient, the things that are here don't want to stay here. They want to move down their concentration or down their osmolarity gradient. So the way we're able to maintain this osmolarity gradient is with a second countercurrent mechanism. And that second countercurrent mechanism is what occurs in the vasa recta. And I have a pretty picture that'll show you that in just a minute. So again, as we reabsorb the water, we concentrate the remaining filtrate, where it can get as much as 4 times as concentrated as it was in the blood plasma, increasing the remaining solutes, the sodium, the potassium, the chloride, which again, as we talked about, then drives that active pumping, and the process continues. And notice, with this decoupling, no longer having solutes and water moving together, what we get is not only do we get a massive reabsorption of water, right? It's about 1/3 of the original volume when it first entered, but it's now also more dilute than it was before as well. And so we have this nice dilute filtrate produced as a result of this. As promised, I told you I'd mention that second countercurrent mechanism that is called the countercurrent exchanger, and this is what the vasa recta does. As I said, the vasa recta basically does 3 things. It brings oxygen to the cells of the loop of Henle so that it is able to do the active transporting that it needs to do. But the other thing that it is going to do is reabsorb all the water that is coming out of the loop of Henle and maintain the osmolarity gradient. And the reason it's able to do this is, notice again, we have a countercurrent mechanism here with this countercurrent exchange. We have blood coming down the vasa recta and then blood coming back up the vasa recta. So we do have them moving in those opposite directions. But the difference between the vasa recta and the loop of Henle is both sides of the vasa recta are permeable to both sodium and water. So if you think about this, on the descending limb, right, what ends up happening is the descending limb picks up solutes. Right? Remember, solutes are going to want to diffuse back down, but what happens is it picks up solutes and brings those solutes down deep into the medulla. And remember, in the descending limb of the loop of Henle, excuse me, this is where all the water is coming out as well. So it's able to help. So as it comes down, so water's coming out, it picks up the solutes, and that helps to maintain that osmolarity gradient. So water's coming out, it's picking it up and bringing it down. The solutes, bringing the solutes deep, releasing the water, maintaining that, and then it picks up the water on the way back up. On the way back up, it picks up the water and it kicks out the solutes, more solutes at the bottom, and less as it works its way up to the top. So basically what we get is this big, huge circular process where the solutes are coming in And the solutes as they enter are being brought down and then come back out later. And water is coming in, but it is being taken out as it goes back up. So so water comes out, but then it is taken back up on the way back up. So basically we have this cyclical process that is taking place, this countercurrent mechanism, where the sodium comes out and is driven down, water comes out and it is brought back up. And in this way, by having this countercurrent mechanism, this circular mechanism of these movements of the solutes and water, we maintain the osmolarity gradient. So again, our descending limb basically absorbs the salt and releases water, but the ascending limb does the opposite. Untitled. And in that way, we maintain that osmolarity gradient. All right. So notice at the end of this process now, like we talked about, when we are leaving, and let's look at the pretty picture, we have produced a dilute filtrate. Again, it entered here at 300 milliosmoles, and I'm not going to write the milliosmoles because it takes up too much space. It gets concentrated to about 1200 by the depths of the medulla. And remember, when it gets back up here to the distal convoluted tubule, it is down to 100 milliosmoles. And remember, as I mentioned, this thick ascending limb and the distal convoluted tubule and the collecting duct are all normally impermeable to water. So notice, once we produce that dilute filtrate, water is not able to leave. Water stays in our distal convoluted tubule, water stays in our collecting duct, and that large volume of dilute urine is released into out the papillae of our medulla into the minor calyx, at which point it is now urine, because it is no longer being processed. All right. And so again, as long as you're well hydrated, this is what's going to happen with your nephron for the rest of the path. All right. However, if you do not have, if you're not hydrated enough, if you do not have enough water in your blood, or your blood volume is down, or your blood pressure is down, or some of those other things that we talked about, then we have the ability to bring more water back. I'm trying to find a place to put this. And again, one of the ways we can tell our nephron that we need to bring more water back is by the use of hormones, like antidiuretic hormone. Antidiuretic hormone was produced where?
Hypothalamus.
There you go. Excellent. But it is released by what?
Posterior pituitary.
There you go. Warms the cockles of my heart to hear you guys. This is lonely doing it this way. I feel silly having the camera on when I'm trying to show the lectures all the time, but I do miss seeing you guys. I do miss the interaction. This is a very weird semester. Yeah, absolutely. It's released by the posterior pituitary. And we talked about from the very beginning when we were talking about this in the endocrine system, that the job of the antidiuretic hormone is to tell the kidneys to reabsorb more water. But now we get to finally talk about how it does that. Basically, what antidiuretic hormone does is it tells the cells of the collecting duct and the distal convoluted tubule to express aquaporins, right? Aquaporins are proteins. As we know, proteins can be kept inside the cell on vesicles. So we have these vesicles, and on these vesicles, we have these aquaporins. What antidiuretic hormone tells these cells to do is basically to take this vesicle, bring it to the plasma membrane, and bind to the plasma membrane. When it binds to the plasma membrane, it puts that aquaporin on the surface of the cell. And when that aquaporin is on the surface of the cell, now water can be reabsorbed. Water is able to be reabsorbed into the cell. All right? What happens when there isn't antidiuretic hormone is the opposite. What ends up happening then, and let's do that, is that what will happen is the cell actually endocytoses. Those aquaporins, bringing them inside. And when it brings them inside, they are no longer expressed. Oops. My eraser did not work. So I'll cheat and just grab it. Once we bring this inside, then water can no longer come inside anymore. So basically, what antidiuretic hormone allows us to do is it allows us to be able to express these aquaporins on the surface. And so that allows the passive movement of water. And if you think about it, in the distal convoluted tubule, remember the osmolarity of our distal convoluted tubule, which is in our cortex, is at 300 milliosmoles. Oops, not 33, 300. Remember here it starts at 100, but if water is able to leave, that water leaving is going to concentrate the filtrate that stays behind, and it could get it as much as 300 milliosmoles, right? Which again, triples the concentration of it, and that is impressive to go from 100 to 300. But remember, this distal convoluted tubule travels all the way through the medullary pyramid. What that means is that it goes through that concentration gradient, which we know at the deepest parts can be as much as 1200 milliosmoles. And so if enough aquaporins are expressed, we are able to reabsorb massive amounts of water in this process, right? Basically 12 times as much water can be reabsorbed as when it first came out of that loop of Henle. And what that's going to do is produce a very small volume of very concentrated urine. So as we know, to control blood pressure, to control blood volume, we use antidiuretic hormone. And now we actually get to see the mechanism by which that works. It's able to express those aquaporins in, excuse me, in our distal convoluted tubule and the collecting duct. Now notice, we talked about how, and I'm going to cheat. Hold on, I need to move this out of my way and this out of my way. Remember, there are aquaporins, and I'll cheat and draw one, on the proximal convoluted tubule, right? We know water is reabsorbed here. We know water is reabsorbed here. That's not what I want. That's what I want. So we know there are aquaporins here and water is being reabsorbed in the proximal convoluted tubule and the distal convoluted tubule. Pardon me, proximal convoluted tubule and descending limb of the loop of Henle. But remember, as we talked about, this was aquaporins that are always present. And so as a result of this, this is what we call that obligatory water reabsorption. Those aquaporins are always there, so water must always follow the stuff and it must always go where we need it to go. Notice with these aquaporins out here in the distal convoluted tubule, and I'll emphasize this again here, in the distal convoluted tubule and in the collecting ducts are only expressed when antidiuretic hormone is present. So notice in this case, this reabsorption of water that's concentrating our urine in this case only occurs with the help of that hormone. And because it only occurs, and I'm going to put that there like that, only occurs with the help of that hormone, we call it a facultative water reabsorption, right? It can only be facilitated when it is helped. It needs help. So again, I'll put this down here where it can be seen a little bit better. There we go. That work? Sort of. I got so many things up on my screen. There we go. Found a place to sneak it in. Excellent. So if you think about it, aquaporins are used for the reabsorption of water, but there are two types of reabsorption of water. There is the obligatory water reabsorption. This is the reabsorption that must occur. It only occurs in the proximal convoluted tubule and the descending limb of the loop of Henle, because those aquaporins are always present. So water must be reabsorbed no matter what. However, out here in the distal convoluted tubule and the collecting duct, here water reabsorption is selective. We only reabsorb water if we're able to express those aquaporins, and we can only express them if we get that help. And that help comes in the form of the antidiuretic hormone. So with the help of antidiuretic hormone, we can absorb more water. So it needs help. And so out here in the distal convoluted tubule and the collecting duct, we have our facultative water reabsorption. All right. It's facultative. It's only helped. It's facilitated by antidiuretic hormone. All right. Questions on that? So there you go. So not only now do we know what's going on in the loop of Henle, but now we know how to produce both our concentrated and our dilute urine. So let's clear that. And lastly, here's that pretty picture from your textbook. Again, notice they've done a nice job of decoupling here to show us what's going on in the vasa recta, to show us what's going on in the loop of Henle. And like I said, we also talked about how we get those differences in producing dilute urine without the antidiuretic hormone. The distal convoluted tubule and the collecting duct stay impermeable to water. So we produce a large volume of dilute urine. But with antidiuretic hormone, our distal convoluted tubule and our collecting duct are now permeable to water, and we get that facultative water reabsorption, which concentrates our urine. So we get a small volume of concentrated urine. All right, questions on that? All right, excellent. All that leaves us with then is the distal convoluted tubule. And right there with the water, we got a huge hint at what happens in the distal convoluted tubule. Again, our distal convoluted tubule, by the time the filtrate reaches there, after passing through the proximal convoluted tubule where most of the reabsorption took place, after going through the loop of Henle where we were able to reabsorb a massive amount of the solutes, we've got that nice dilute. So notice we only have about 10% of the original solute remaining and about 25% of the remaining water. Now it is at this point really what we could call almost urine, right? Again, that's not a real term, but remember, as we talked about, this almost urine is what is being checked in the macula densa, because it's mostly done. At this point, what we need to do is fine tune it, right? An example of that is our blood volume, our blood pressure. If we don't have enough water in our blood, if we don't have a high enough blood pressure, we released a hormone, antidiuretic hormone, that told the kidneys to bring back more water, right? We were fine tuning based on the condition of the blood. And we use hormones to tell the distal convoluted tubule to do that. And it's not just water. Other hormones tell our distal convoluted tubule what to do as well, right? Remember we talked about aldosterone. It's the most common mineral corticoid, produced by the zona glomerulosa of our cortex of our adrenal gland. And being the most common mineral corticoid, it tells us to monitor the conditions of our most common ion, our electrolyte, sodium. So if we don't have enough sodium, aldosterone tells the kidney, reabsorb more, right? And again, where sodium goes, chloride tends to follow. Potassium is also typically secreted, and we see that here. This is not a pathway you need to know, but in the distal convoluted tubule, but it's a pretty picture from your textbook. Remember we talked about parathyroid hormone. Remember parathyroid hormone tells the kidneys to reabsorb calcium. So we get that calcium reabsorption that occurs here with that. And as we talked about, antidiuretic hormone, we already talked about the reabsorption of water. So these are plenty of examples of hormones that we've talked about that are telling the distal convoluted tubule, and to a lesser extent, our collecting duct to reabsorb these things to, again, we don't care about the condition of the urine, we care about the condition of the blood. So we are looking at the blood, seeing what the condition of the blood is, and using these hormones to modify that. All right. So in this way, we're able to regulate pH, ion levels, right? We can get sodium back, we can get water back, we can get bicarbonate back, and also secrete hydrogen ions. And this can also be where we're going to be able to get rid of some of those large wastes and toxins via both diffusion and active transport. Urea, again, is constantly being pumped out here, and other things along those lines as well. And again, none of those are specific pathways you'll be responsible for. Lastly, remember, once the, whoops. There we go. Once the filtrate leaves the distal convoluted tubule, it has exited the nephron, right? So once it reaches the collecting duct, when it reaches the collecting duct, it has left the nephron. But it's still filtrate because it can still be processed. And as we talked about, our collecting duct basically has two types of cells. These two types of cells can still process the filtrate, and so that's why it's still called filtrate. And those two types, as we talked about first, were a principal cell. Here we see an example of a principal cell here. Notice one of the primary functions of the principal cell is the reabsorption of sodium and the secretion of potassium. These are also the ones that will express the aquaporins. The picture doesn't show this, but with antidiuretic hormone, these are the ones that will express aquaporins with the help of antidiuretic hormone. So again, they play a role in that facultative water reabsorption. And even though I don't have a picture of it, we also have our intercalated cells. The intercalated cells are primarily involved in maintaining the pH of the blood. So they play an important role in the secretion of hydrogen ions and the reabsorption of bicarbonate. And it uses the exact same mechanism we saw in the proximal convoluted tubule for that. But it's going to be, again, this is hormone driven, where we're going to get that expression. And then also, again, we do want to absorb some potassium. So as a net effect of this, there is some reabsorption of potassium that takes place in that intercalated cell as well. All right, and so then, like I said, it is going to secrete those hydrogen ions, bring back the bicarbonate, play an important role in our regulation of the pH of the blood. And again, here is that summary figure. Like you said, your book's got a great in focus that does a nice job of showing, clear all of that, those pretty pictures, putting all the pieces together. At the end of all of this, excuse me, that collecting duct finally expresses the filtrate out into the minor calyx. When that occurs, it is now finally urine. While we started with 200 liters, 180 to 200 liters of filtrate being made during the course of the day, we end the process with about 1 to 2 liters of urine. That urine at this point is mostly water, about 93 to 97%, and pretty much nitrogen-based wastes. Typical urine has a light yellow straw-like color that comes from the pigment urobilin. We didn't do digestive yet, so one of the things we need to talk about is that one of the things that our liver does is it breaks down red blood cells. Right, we also talked about the spleen doing that, but the liver also has an important job in doing that. And when it does that, it breaks down the hemoglobin. When it breaks down the hemoglobin, that process produces a pigment called bilirubin. Bilirubin plays a role in jaundice when those levels get too high and things along those lines. But bilirubin is used by the liver to produce bile. And that bile plays an important role in the breakdown of fats. And we'll talk about that, the emulsification of fats. We'll talk about that in the next couple of weeks when we're talking about the digestive system. But what's important for us is that bilirubin in the digestive system is broken down into 2 pigments. So bilirubin is broken down into 2 pigments, and those are stercobilin and urobilin. Stercobilin is actually a brownish pigment. It actually stays in the digestive tract and is the primary colorant of your feces. So the brownish coloration of your feces is in large part due to the breakdown of red blood cells by your liver to produce that bilirubin, and that bilirubin gets broken down. That bilirubin also gets broken down into urobilin, but that urobilin is actually reabsorbed by the digestive system, and it is released by the kidneys. And so that yellow coloration of the urine also comes from the breakdown of red blood cells. Now, there are other things that can affect the coloration, the concentration. The more dilute, the clearer, the whiter the urine is, whereas the more concentrated, the darker that pigmentation is. Certain foods, certain medications, things like that can also change the color of your urine as well. Now, let's go ahead and get rid of that now. Your urine, healthy urine, should be clear. There should not be a cloudy particulates in that. If that occurs, that can be an indication of, for instance, urinary tract infection or something along those lines. Urine is slightly more dense than water, right? While you're at home looking for fun ways to play with your anatomy and physiology, one of the things you can do is urinate into your toilet and then don't flush. Wait a couple hours and come back, and what you should see is that the urine will actually condense down at the bottom of the bowl, right? When you first void the urine, really urination is not the appropriate term. Urination is the formation of urine, and we're doing that all the time. Micturition is the correct term. But when you void that urine, it mixes with the water and the whole bowl becomes yellowish in color, but if you let it sit, the urine will typically sink to the bottom because it is slightly more dense than water, so they will actually separate out. It is sterile. Now, again, this is usually when I make a joke when we walk down downtown Sacramento, right? That smell of urine hardly makes you think sterile. But the important thing to remember is that when the urine is first produced, it is sterile. However, it's water, it's warm, and it has a lot of nitrogen-based wastes to it. Just the kind of thing that bacteria absolutely love. Excuse me. So what ends up happening is you express the urine, you void the urine, right, on that building in downtown Sacramento, and bacteria then come to that and feed off of that, and it is the bacterial wastes that are produced that give downtown Sacramento its distinctive aroma. So like I said, I know we don't think of sterile when you're first at, although by this point, everybody's gone from downtown Sacramento. It may still smell wonderful now. I have no way of knowing. Lastly, again, the pH of our urine can vary dramatically. For the most part, it tends to be acidic, around 6 pH, because again, remember one of the things we want to do is hold on to that bicarbonate, and we do that by expressing hydrogen ions. So typically it is more acidic than our blood and inside. However, our diet plays a huge role in the acidity of our urine. If you tend to be more of a carnivore, it can be incredibly acidic, actually down to as much as 4.5 pH. However, if you have a strictly vegan diet, which I know nowadays is super hard to do, but if you were a strict vegetarian, in some cases, your urine can actually become slightly basic from that. So a diet can play a major role on that as well. And the same thing for the mild aroma associated with it as well. There are certain chemicals, foods, things like that that can change the consistency of it as well. All right. Questions on that? So there you go, slightly aromatic. But again, those bacteria can change that. Typically when first produced, it shouldn't have a pungent aroma, although diet can change that, certain medications, and then obviously some medical conditions can affect that as well. All right. Now that we have made our urine, all we have to do is move and store it and release it. So this is a good point for our next second break. So let's go ahead and take our second break. We'll take another 10-minute break. It looks like it's 10:17 now. So we'll come back at, 10:07 now. So we'll come back at 10:17. All right, oops. Any questions before we take our break? All right, excellent. Then I will see you guys in 10 minutes. So, like I said, our heavy lifting is done at this point. Our goal now is to basically, because we're constantly producing urine, constantly filtering the blood, now we just need to talk about the process by once we're done making it and processing it, we store it and then release it from the body. And that is the job of the urinary tract. As our illustration here clearly shows, and let me get my annotation tools so I can get my little spotlight. It is comprised of the two ureters. So we have two ureters here, one bladder and one urethra. And I know ureter and urethra are similar sounding terms, but what I always remember is ureter we have two of them and there are two E's in it, whereas there is just one urethra and only one E in urethra as well. So that's something that helps me to be able to remember those. Let's talk about the ureters first. The ureters are what exit out of the kidney. So again, if you think about it, it is that renal pelvis that is that funnel-shaped structure. In fact, if we want to really be specific, if we think of the path, it goes from the minor calyx. Oops, hold on one second. Forgot to close my office door, and my kids are tired of listening to me talk. from the minor calyx into the major calyx, or calyces, if we're going to go plural, but we'll go singular. And from that into the pelvis. And then that pelvis is that funnel-shaped structure that then leaves the kidney and it feeds into the ureters. The ureters, like the kidneys, are retroperitoneal, behind the peritoneal, the parietal peritoneum in that fashion. And their job is to move the urine from the kidneys to the bladder. Now, because if you think of the orientation of this, right, downward, you would think that gravity should be sufficient to be able to move urine through them. However, right, if you're laying in bed or if you're doing a handstand, you don't want the urine slowly feeding back into the kidneys. And definitely, if you're an astronaut in outer space where there is no gravity and you're on the International Space Station for 6 months, you'd want to probably urinate once or twice while you're up there. So it is actually going to move the urine with rhythmic contractions of the smooth muscle, which of course we call peristalsis. So we're going to use that peristalsis, that rhythmic smooth muscle contractions, to move urine from the kidney into the bladder. One of the interesting things in here, let me clear this and do a simple drawing. If this is, and let's cheat and make it solid. If this is the bladder, one of the things that our ureters do is they come in. So again, let's orient ourselves. This is the anterior part of the body. So this is a sagittal view. What the ureters actually do is, rather than attaching directly to the front, the top, right, which makes sense, it doesn't do that. What it does instead is it goes posterior to the bladder and actually attaches from the posterior and the inferior side. Now, it might not make a lot of sense as to why that would be the case, but if we change our drawing a little bit, let's go ahead and clear that. Here is our bladder. I'm just going to draw it as a block for the kidney, although that's not obviously the case. If you think about it, if the ureter came straight down into the bladder from the top, as the bladder expands, as it fills with urine and gets bigger and bigger, what would end up happening is that it would push the ureter upwards. And one of the problems with pushing the ureter upwards is it could cause the ureter to kink. And obviously, if we kink, like if you kink the hose, that stops the flow of urine into the bladder and instead causes that urine to back up into the kidney. And if it backs up into the kidney, we can get a dramatic increase in pressure inside the kidney that can be very damaging and potentially cause problems. This is similar to that process of ptosis that we talked about that could be an issue as well. So instead, one of the advantages to having the ureters, coming down and into the bottom is it allows for the expansion of the bladder without kinking the urethra. The other thing that happens is as this fills, there is going to be an increase in pressure. Yes, we have our transitional epithelial tissue in there that allows for the expansion of the tissue with a decrease in the pressure that is going to be produced by it, but there's still going to be an increase in pressure. And again, we don't want that pressure backing up into back into the kidneys. So if you think about it, as it fills, what ends up happening is the pressure of it pushes down on the opening of the ureter, and as a result of it, it pushes it closed. So basically what happens is as the bladder fills, it closes the opening. It collapses the opening of the ureter? Well, but if the opening is closed, then how can we possibly get urine into there? Well, remember how the ureter is doing it. It uses peristalsis. And so when it contracts, it forces the opening open, and urine enters. And then once that peristaltic wave stops, then the pressure of it collapses it again, and it stops the backflow. So really that's the take-home message. This orientation stops the kinking, and it stops the backflow of urine, both of which protect the kidney. So that's really our goal there. Our goal here is to protect the kidney, and the location of the ureter helps us to be able to do that. Now, one of the things that you're definitely responsible for on this exam is histology. Hopefully you've been looking at those things. The histology for all of these structures are similar because remember, starting at the minor calyx and then working down the entire urinary tract, it is all going to be lined by a transitional epithelial tissue. Transitional epithelial tissue, as I'm sure you remember, is multiple layers of cells, right? It is a stratified tissue. However, typically the cells get larger as they go towards the surface. So we start with smaller cells at the basal surface, and as we move to the apical surface, the cells get larger. And as you can see, we have these large block-like cells, brick-like cells on the apical surface, right? We want a solid barrier because remember what's in here is urine, and there's nothing useful in that. We don't want any reabsorption to take place. So we want that urine to stay in there. So we want this big protective barrier, and that's what we get with this transitional epithelial tissue. But remember, transitional epithelial tissue is transformative. It can expand without elasticity. So it's able to stretch and not get a dramatic increase in pressure. Of course, this is still part of a mucous membrane. So of course, it sits on top of a lamina propria, which of course is the name of the tissue based on its location. And what type of tissue is the lamina propria again?
Mucous membrane or
Well, it's part of a mucous membrane.
connective areola.
There you go. Areolar connective tissue, absolutely. It is an areolar connective tissue, and based on its location, it's a lamina propria, right? Any areolar connective tissue that is part of a mucous membrane, we call it a lamina propria based on its location. And then we have the muscularis on the outside. And notice with the muscularis, it's essentially just kind of a single layer of smooth muscle so that we're able to make those contractions that are going to be able to, again, those peristaltic contractions to move the urine through that. Now, our goal, as we're going to see, the ureter not only spells similar to the urethra, but looks similar to the urethra. So there are some big key differences. The first is the size. Right? Notice the ureter is much, much smaller. So we can get it in view and still be able to see the cells pretty well. If we're going to get the entire urethra, when we look at the entire urethra, you'll see that it is going to be a much lower magnification view to be able to get all of it into the field of view at the same time. Another big difference, as we said, is the muscle layers. This typically just has one muscle layer to it, whereas the urethra is going to have two, an inner circular and an outer longitudinal. So we will see that as well. Notice here you get kind of a collapsing of the transitional epithelial tissue, but you'll see in the urethra, there's going to be much more invaginations. So again, the size is going to be one, the folds and the lumen, they're going to be much more excessive in the urethra, much less excessive here in the ureter. Smooth muscle layers. typically 1 here in the ureter, 2 in the urethra. And the last is the lamina propria. The urethra is basically the opening to the outside world. So it has to protect us. And one of the ways it does that is by having a larger lamina propria, a much larger lamina propria with big mucus producing glands, because after all, the mucous membrane. So we're going to see a much more glandular and a larger lamina propria. So those are the things that you're going to look for to distinguish the urethra from the ureter. And we'll take a look at that when we see the urethra later. So this is the ureter. We will take a look at the urethra in just a moment. All right, questions on that? All right, before we can talk about the urethra, though, we have to talk about what's in between. And what's in between is the bladder. The bladder is similar to the stomach. And again, I know we haven't talked about the digestive system, but basically it is a collapsed muscular sac, right? And again, you go to the store, you have to bring your own bags now, right? And so you take that bag and you can crumple it up into a ball so that it fits into your pocket. And then you pull it out and it is able to expand. Basically, it has those folds that allow it to expand, to enlarge, and fill up the space of all of your groceries. Well, the stomach and the bladder work the same way. They're able to collapse down into this tight, compact structure that has all these folds inside. And these folds inside are called rugae. And that's what they're trying to emphasize with these undulations that we have here. We have that ability for that bag to expand because of all the folds. One of the important things to remember is these rugaes are formed by the mucosa, right? And the submucosa, but not the muscularis. So there are parts of the wall that come off and form that. There is a smooth muscle layer. So we have the inner surface, which is the mucosa. We have the middle layer, which is our smooth muscle layer. And again, we didn't talk about the digestive system, but one of the things when we get to the stomach we'll learn is that the stomach has 3 layers of smooth muscle. However, in the stomach, those 3 layers are very precise. There is a circular layer, there is a longitudinal layer, there is an oblique layer. And all of the cells are kind of arranged in 3 distinct layers. Our bladder has 3 layers as well. But the job of the bladder is to contract, kind of like the heart was. And if you remember, our muscle fascicles in the heart were kind of circular or figure 8 shape. And we see the same thing in the bladder as well. And because of that, the 3 bladders are not, the 3 layers of smooth muscle in the bladder are not distinct. We'll look at it histologically in just a moment here. And while you'll see lots of fascicles, lots of bundles of muscles, you're not going to see 3 precise layers. So instead, what we end up having, I'm sorry? So instead, what we end up having is just a collective muscle. The 3 layers of smooth muscle are just collectively known as the detrusor muscle. So we have that detrusor muscle that is basically made up of those 3 layers of smooth muscle. Ball shape, figure 8 shape fascicles to again constrict and collapse the bladder to force the urine out. When we look at the anatomy of the bladder, notice there are 3 openings that can be found inside the bladder. There is the internal os, or internal opening of the urethra, and there are the 2 osses, or openings, and again, I keep using that term, so make sure you know it. Os is the name of an opening. We have the 2 osses, or openings, for the ureters. So here we have the 2 urethral openings, and we have the internal os of the urethra. There's also an external opening, an external os of the urethra as well. Notice in between those three openings, we have a triangular shaped region, and this triangular shaped region is what is known as the trigon. In this area, the trigon is a thickened muscular wall in there. The mucosa is thickened as well. It makes it a little more rigid. The advantage of this is it keeps these openings from collapsing and gives us that pocket where the urine can accumulate and then expand out from. However, this can also be important from a clinical standpoint, because when it comes time to void your urine, you're not going to be able to completely empty your bladder, right? It's like that can of soda. No matter how much you shake it, no matter how much you tap it, there's always going to be a little bit left inside. And the same thing is going to be true for the bladder. The urine that remains inside the bladder accumulates in this trigon region. This becomes significant because, as we know, urine is warm, urine is moist, urine has a lot of particulates, you know, a nitrogen-based waste and things along those lines. So this can be a place where infection can occur. So many bladder or urinary tract infections start here in the trigon, in that urine that is remaining here in the bladder and is not fully expulsed. As we mentioned, and let's highlight it again, we'll use blue this time so it stands out. This entire region, as we mentioned, of smooth muscle is the detrusor muscle. However, at the base of the urethra, where the internal urethral os is located, what ends up happening is we get a thickened circular muscle. This thickened circular muscle basically forms a valve. Whoops, wrong thing. Hold on. Gotta go back. That's what I want. This valve basically controls one of the two valves that controls the flow of urine out of the urethra. And this one that is an enlargement of the detrusor muscle, enlargement of that smooth muscle, is known as the internal urethral sphincter. Now, again, as I mentioned, this is comprised of smooth muscle. So this is not something we can voluntarily control. This is involuntarily control. And specifically, it is going to be controlled by the parasympathetic nervous system. Right, so we have that involuntary control of smooth muscle, and so we have the smooth muscle valve that is going to be controlled by our parasympathetic nervous system and our reflexes. And we'll talk about that in just a minute. So let's clear all of those. And there you go. Here we look at the histology. Now, obviously, this is a human, excuse me, a human bladder. So a human bladder is not going to fit on a single microscope slide. Notice as we look at it, it is such a low magnification. We cannot see really the individual nuclei of the cells of that transitional epithelial tissue, but we can see that transitional epithelial tissue. We can see the lamina propria and even a submucosa under it. And notice these big, huge folds in it that we see. Those are those rugae that we talked about, the big that are going to allow it to expand. Those are the collapsed bag of that plastic bag. Here we see the detrusor muscles, and here we see a fascicle, and here we see a fascicle, and here we see a fascicle. But again, notice it's not three distinct layers. So we don't have to worry about identifying the individual layers of them. We don't have individual names, just all three smooth muscle layers together collectively form what we call the detrusor muscle. That big, powerful muscle that is going to collapse the bladder to help to express the urine. And of course, being an intraperitoneal organ, it's in the abdominal pelvic cavity. The outer surface is a visceral peritoneum. And of course, we know that visceral peritoneum, like all serous membranes, or serosas, so let's do it that way, is comprised of a simple squamous epithelial tissue on top of an areolar connective tissue. And this areolar connective tissue is not a lamina propria, right? Only this one is because it's part of a mucous membrane up here, the mucosa up here. However, as we do know, a simple squamous epithelial tissue that is part of a serosa, based on location, we call it a mesothelium. So there you go. So the areolar connective tissue doesn't have a name in a serosa, but the simple squamous, the epithelial tissue, does. And so that lines the outer surface of our bladder. Alrighty. That leads us to our urethra. Our urethra is, again, a muscular tube. Its job is to carry the urine outside the body via peristalsis. And as we talked about, again, our bladder is going to contract. It is going to produce force. I know when you first start urinating, or micturating, which is the correct term for the voiding of our urine, the flow is continuous. And that continuous flow is from the high pressure that is being produced in our bladder from those contractions. However, as the bladder starts to empty, one of the things that you'll notice is that the flow of urine becomes more pulsatile in nature. And that pulsatile nature of it at the end is from those peristaltic contractions that are helping to expulse the last bit of remaining urine from the bladder. So it does use peristalsis as well as the powerful contractions of the bladder for that release. Here we see the histology of that urethra. So again, notice, as we said, its size is much, much bigger. Here we can see the entire lumen of the urethra in our field of view, but notice we can't see the individual cells. Because this is such a larger structure, we have to look at a much smaller magnification. So that was one of the big differences we mentioned between the urethra and the ureter. Notice the size of the lamina propria is much, much larger in the urethra. And you can actually see that there are some large glands, mucus-producing glands in here. Notice also we can see two different orientations to the smooth muscle. Notice this smooth muscle is more longitudinal in its view, and this is more cross-section. So we have those two layers of smooth muscle there as well. So like I said, those are the things that you're going to want to look for to really distinguish the difference between the urethra and the ureter histologically. They are similar because they're both muscular tubes to carry urine, but there are two or three major differences between them that should be easy to distinguish. The other important thing about the urethra is the urethra, and again, in the lamina propria also a lot of elastic fibers in there as well for expansion with those mucus glands. But the other big difference between the urethra is the first structure that we've talked about in the class, really, where we see significant differences between males and females. I mean, you can argue we talked about differences between the testes and the ovaries, but right, males and females have one or the other. You don't have both. Both males and females have urethras. Both males and females have hearts. Both males and females have lungs, right? While there are statistical differences in the hearts, right? Male hearts are slightly larger than female statistically. And again, as we've talked about, that's because we've had billions of humans to measure the size of their hearts for. So even small numbers can become significant when you have that large of an N. But as we also talked about, those differences aren't meaningful. Here, for the first time, we have meaningful differences in an organ between males and females. The urethra in the male and the urethra in the female are very different in their anatomy and in their function. Let's talk about some of those differences. One of the big differences between the urethra in males and females is length, right? Males have the longer urethra. In females, the urethra is only about 3 to 4 centimeters in length. So if you think about it, from the bladder to the outside of their body, it's only 1 inch. This is one of the reasons why bladder infections are much more common in females than they are in males, because in females, the bacteria have a much shorter distance to have to travel to get to the bladder. Whereas in males, the urethra, notice we're talking urethra length, we're not talking penis length. The urethra length is about 8 inches. Locations, obviously, as I just mentioned, the males, the urethra passes through the prostate and through the penis as it exits the body. Whereas in females, it does not actually enter the vagina. It runs parallel to the vagina and is actually anterior to it. So it is along the outer wall of the vagina, but does not actually pass through it, where it does pass through the penis in males. This is significant for a functional standpoint then. In females, because their copulatory organ, the vagina, and the urethra are separate openings, they have separate functions. So in the female, the urethra is only a passageway for urine. However, the males, because the urethra goes through the prostate and goes through the penis, it is part of both the urinary system, but also the reproductive system. And so in males, that urethra carries both urine and semen, whereas in females, it just carries urine. And in females, the urethra is only a urinary organ. In males, it is a urinary organ and a reproductive organ. And that's basically because of its location of passing through the penis. Here we've done it with the pretty words. We can do it here with the pretty pictures. Here we see the female. Let me switch back to my highlighter. Again, there's our detrusor muscle. There's our transitional epithelial tissue. Here's our trigon. Here is our internal os, or opening of the urethra, and the external os, or opening of the urethra. And it basically is a single inch long pathway anterior to the vagina. But one of the important things to remember is it does have to pass through our urogenital diaphragm. Does have to pass through the urogenital diaphragm. Hopefully you remember the urogenital diaphragm from when we were in 430 and we talked about the muscular system. It helps to support the pelvic organs. It plays a role in both defecation and urination, or micturition more appropriately. And as you hopefully also remember, the primary muscle is really a muscle group made up of the iliac coccygeus and the pubic coccygeus, or what collectively we call the levator ani muscle. Contracting the levator ani muscle helps to provide strength and support for your urogenital diaphragm. And again, if you are about to give birth or have recently given birth, there are those Kegel exercises that where you contract that muscle, hold it for a count of 10, relax it, do that in sets of 10. And those Kegel exercises help to strengthen that levator ani. So notice our urethra has to pass through that. Notice also as it passes through it, and I will highlight it this way, there is an enlarged circle of muscle, an enlarged circle of that levator ani that we call the external urethral sphincter. Oh, wait, no, that's not it. Hold on, where is it? There it is. Here. Our external urethral sphincter. Now notice this is an enlargement of our levator ani muscle. This is an enlargement of the urogenital diaphragm. So this is skeletal muscle. Which means we can voluntarily control. And then obviously reflexes as well. So notice we have not one, but two sphincters that are going to play a role in our release of urine. We have an internal urethral sphincter that, as we talked about, is comprised of smooth muscle and we have our external urethral sphincter that is comprised of skeletal muscle. And notice they're separated from each other. The internal urethral sphincter is part of the detrusor muscle, whereas the external urethral sphincter is part of the urogenital diaphragm. All right, so notice we have those two sphincters, two different anatomies to them. All right, and I think I, in fact, emphasized that there and there. All right, so the external sphincter we have there, and then we have the internal as well. All right. By comparison, here is the males. Notice, as we mentioned, the males has to travel through the prostate, which is an important gland in the reproductive system we'll talk about, and it passes through the copulatory organ, that is the penis. As such, the urethra being much, much longer, and anatomists being what they are, they of course love to name everything. So in the urethra, in males, right, has three distinct regions. So the organ in the male has three distinct regions. All right, how many distinct regions to the females? None. It's just the urethra. One reason, because it's shorter, and second, because it's females and anatomists don't care about females. All right, so if we look at the male urethra, basically there are three distinct regions, and we distinguish them by the structures they pass through. All right, so notice the first part we call the prosthetic urethra. And guess what it is that the prosthetic urethra passes through? Well, passes through the prostate. All right, there you go. That's pretty simple. The second region is the membranous urethra. The membranous urethra is where it passes through the urogenital diaphragm. And then lastly, we have the spongy urethra, what is also called the cavernous urethra, or also the penile urethra, and that is where it passes through the penis. So we have three distinct regions to the urethra. So our urethra has three distinct parts. Again, kind of if you think like the pharynx in our respiratory system, had three distinct regions, each associated with a different opening. So the nasal pharynx, the oral pharynx, the laryngopharynx. Well, here we have the prosthetic urethra, membranous urethra, and the spongy urethra. Now, while there are these big morphological differences in females, there are some things that are not different. One of those things that are not different is, again, we have that enlargement of the detrusor muscle. And that enlargement of the detrusor muscle is that internal urethral sphincter. And again, this is going to be smooth muscle. Excuse me. Smooth muscle, like we talked about, has that involuntary control there. And once again, we have a thickening of the urogenital diaphragm, and that thickening of the urogenital diaphragm is our external urethral sphincter. Which again, being part of the levator ani muscle, is skeletal muscle. And so of course we can voluntarily control it as well. Notice there is a bit more urethra between them. The prosthetic urethra basically sits between them. But they're still offset from each other, just like in the female. And just like in the female, we have an internal and external urethral sphincter. Internal is smooth muscle, part of the detrusor. External is skeletal muscle, part of the levator ani. All right, questions on that? Oh, and here's another view for the male. And again, I won't draw on it all the same, but again, notice through the prostate, coming through, prosthetic urethra, through the levator ani muscle. So that right here, this tiny point right here is the membranous urethra. And then all of this through the penis is the spongy urethra. So we see that anatomy. And again, we have those two sets of sphincters. Right, we have the external sphincter there, and again at the base of this we'll have the internal there as well. We keep making a point of emphasizing this. Oh, and here we see the sagittal view, just so that the females don't feel left out. We see the same thing. What's nice about this picture is we see the relationship of the urethra to the vaginal canal. Here is the vaginal canal, which brings you up to the uterus. Right, this is where you grow the basketball for 9 months. But the vaginal canal's opening and the urethral opening are separate. Notice both have to pass through the urogenital diaphragm. And of course, where it passes through the urogenital diaphragm, we will have that external urethral sphincter. And up here at the internal os of the urethra is where the internal urethral sphincter will be located. All righty. Questions on that? All right. All that leaves us with, the last thing we have to talk about then is the voiding of our urine. As we mentioned, we are constantly producing urine. As we are constantly producing urine, we need to then accumulate it, get it to the appropriate, then get to a socially appropriate location and void it. So I'm going to go through and write it all out. I know I'm going to draw it here. I know this doesn't necessarily help you guys for the essay questions because I know you guys can't draw them anymore, but I still like my drawings. So there is our bladder. Here is the urethra, and then we'll use that. For the urogenital diaphragm. All right, now we need to put our sphincters in here. So we know up here we have the internal urethral sphincter, part of the detrusor muscle. And then down here we have the external urethral sphincter as part of our urogenital diaphragm, as part of that levator ani muscle. So let's label these. So this is our internal urethral sphincter. And again, as we know, this is smooth muscle. And then this one down here is our external. And it is skeletal muscle. Excellent. All right. This reflex basically uses micturition reflex, uses both local, well, let's say it this way, I like it this way, intrinsic and extrinsic reflexes. All right. We might need a little reminder of that. Intrinsic means within the organ, and extrinsic means that it involves our nervous system, our central nervous system. Oops. And as I already mentioned, that involvement of the central nervous system, I'll give myself a little bit more room, involves the parasympathetic nervous system. And so I'm going to go ahead and abbreviate that as PSN because we already talked about that. Smooth muscle controlled by the parasympathetic nervous system. All right, excellent. So obviously, what we need to start this reflex, like all reflexes, is we need some type of stimulus. We need some type of trigger. And that trigger basically comes from the stretch of the bladder. What happens is our bladder fills with urine. And as it fills with urine, our bladder expands. As our bladder expands, our bladder stretches. And I don't like that. Let's use black. We get this expansion of the bladder. In our bladder's wall, we have these stretch receptors. It's not a new concept, something that we've talked about again. They produce action potentials based on length. And when they stretched, they produce more action potentials, right? That's how our stretch receptors work. When it stretches, it produces more action potentials. So this is our trigger. That trigger is the stretch of the bladder, and we get that increase in production of action potentials. That stretch of the bladder, that increase in action potential, basically causes two reflex arcs. One local and one long, right? One intrinsic, one extrinsic. Let's talk about the short reflex first. So let's be specific. Short, right, which is again our intrinsic, and one long, which is our extrinsic. Let's talk about the short reflex first. All right. With that short reflex, what happens is those stretch receptors send a signal to the detrusor muscle. So these stretch receptors, they send a signal to the detrusor muscle. And what happens is the detrusor muscle starts to contract. That's the first thing that it does. But it does two other things as well. So again, it causes this detrusor muscle to start to contract. And obviously, if we're starting to contract, we're increasing the pressure in the bladder. All right. So that is the short local reflex. However, the other thing that happens is it produces that long reflex. Right? Through the parasympathetic nervous system, we trigger two events that are going to occur in our urinary system. the first thing that is going to happen is our parasympathetic nervous system is going to relax the internal urethral sphincter, right? So again, those stretch receptors are going to send a signal to my peripheral nervous system, and that peripheral nervous system is going to send a signal to this internal urethral sphincter, causing it to relax. This will allow a little bit of urine between the contractions that are happening in here and here. Will allow a little bit of urine to enter into the urethra. And you can feel this. You have tactile sensors in here, right? And so this is when you start doing the pee-pee dance, right? You can feel the urine enter the urethra. You can sense that, and that's when you realize and know you feel the need to void. Between the contractions of the bladder and the feel of the urine in the urethra, you feel the sensation that you're going to have to void. However, when you feel that sensation, do you void instantaneously right away? No, and the reason for that is that that long reflex does a second thing. It causes a contraction of the external urethral sphincter. So the same time that it is relaxing this sphincter, it is causing a contraction of the external urethral sphincter. All right. This muscle is a muscle that requires strength. We are not born with the strength in this in this external urethral sphincter to stop the flow of urine, which is why when a newborn baby's urine contracts an internal urethral sphincter, what happens? Urine comes out. All right. That's why we put them in diapers. One of the first big events is being able to learn to control that external urethral sphincter. However, on the flip side, one of the reasons grandpa is also in diapers is because as we age, this external urethral sphincter, that skeletal muscle, weakens. And again, now it's no longer able to keep that urethra closed. And so when that urine is expressed into the urethra, grandpa can't stop it from coming out either. So that's why your baby and your father are both in diapers, because of, again, weak external urethral sphincters. So this contraction here stops the urine from coming out, but ultimately, eventually, you are going to want to void. So you get to a socially appropriate location. And once you get to that socially appropriate location, because it is skeletal muscle, you voluntarily relax. Excuse me. You voluntarily relax the external urethral sphincter. And when you voluntarily relax them, both sphincters are now open. And voiding occurs. All right? Just like we want it to. Questions on that? All right, so let's understand this process. And one of the ways we can understand the process is to see how things change. One way these things can change could be that when you feel that need to void, because the urine, your bladder is contracting and the urine is entering your urethra, you're in your car driving and you just passed that sign that said next rest stop is 30 miles away. All right. Now, again, you have two choices at that point. You can pull over to the side of the road and find a bush, or you can hold it. And what happens if you hold it, then eventually, and this is a term we've used before, eventually what happens if you hold it, if you refuse to release, what happens is we habituate to the signal. And as we habituate to this stretch of the action potentials from here, then what ends up happening is that that signal goes away, the bladder stops contracting, and your internal urethral sphincter constricts again. All right? That's a problem because now you've gone the 30 miles, you finally get to the rest stop, you get out, and you have trouble going because now that urethra is not open anymore. Or maybe instead you, again and again, this is an example that who knows if it will ever be relevant again for two reasons. One, because the Raiders have abandoned California for Vegas, so you can't go to the Oakland Coliseum anymore. And more importantly, you're not allowed to be by more than 3 people at any one time now. But back in ancient times, there was a team called the Oakland Raiders who played at a dump of a stadium known as the Oakland Coliseum. One of the fun characteristics of the Oakland Coliseum, and again, I can't speak to the girls' bathroom because I was never in there, but in the boys' bathroom, basically you had troughs that you would stand around and urinate into. And so one of the things that can happen, some individuals have what they would call a quote-unquote shy bladder, right? When you're exposed to other people, you have difficulty urinating. The reason for that is this right here. It is the parasympathetic nervous system that relaxes that internal urethral sphincter. If you are stressed or nervous, that is a sympathetic reaction, and that sympathetic activation suppresses the parasympathetic, causes trouble in the relaxing of the internal urethral sphincter, and voiding can become an issue. Now, you're driving, you hit the 30 miles, and now you hit 30 miles and you don't feel the need to void anymore, so you keep driving. And you get 15 miles down the road, and the problem is the need comes back. While the signal will habituate for a while, what happens is that the bladder continues to stretch. And as it continues to stretch, the result of that, is that the signal becomes bigger. And the contractions become stronger. So while you can hold it for a while, as you continue to hold it more and more and more and more, then what can happen is that the contractions become stronger and stronger and stronger, bigger and bigger and bigger. And at that point, there is the potential where the contractions of the bladder can become so forceful that it can actually force that external urethral sphincter open, and you're voiding your urine whether you want to or not. All right. One last thing that I will mention, because many of you are hopefully still thinking of going into the medical fields. The other issue that comes with for this for many nurses, and again, it's less of an issue now, but is this habituation to the signal. Back in ancient times, and by ancient times, I mean as recently as 4 or 5 years ago, nurses were not good about getting breaks on a regular basis. They could do an entire 8-10 hour shift and never have the opportunity to go to the restroom. And so they're constantly having to hold the urine, constantly having to avoiding. There you go. Didn't mean it that way, but that sounds fun. And so as we mentioned, what happens is you habituate to these signals. And there's been several extreme cases where a nurse has had to hold their urine so long that basically they've completely desensitized their micturition reflex. And so now what ends up happening is that they're not capable of voluntarily voiding anymore and actually have to use a catheter to be able to expel urine from their bladder. Because by holding it for so long, for such a long period of time, for such many days, weeks, months, years, their system has become so habituated, so desensitized that they can't void normally and actually have to use a catheter to be able to void urine from their bladder. All right. Fun stuff. All right. Questions on that? I like that. I'll take a picture of this. Perfect. Oh, hold on. Let me move this out of the way there. Do that again. Excellent. Any questions on that? All right. With that, that is everything we have. Let me go ahead and clear this. We can go ahead and look at the pretty pictures of this again. Again, our bladder fills, stimulates the stretch receptors. We have that sacral spinal reflex, again, regulated by the parasympathetic nervous system, where we basically get three big effects. We have a local reflex where it causes our detrusor muscle to contract. All right, this is our local intrinsic effect. And then the relaxing of the internal sphincter, the contracting of the external sphincter is part of that extrinsic reflex. We then voluntarily relax the external sphincter and voiding occurs. If not relaxed, the contraction will stop, but again, it will continue to fill. And again, if the bladder fills to about 400 milliliters, the contractions of the bladder can become powerful enough where they can actually force that external sphincter open. All righty. Questions on that? All right, well then just that simply, that is everything you are responsible for the urinary system. All righty. So that's it, folks. That's all we have to do. Respiratory system, urinary system, test on Monday. That is all the new material that we have. Is there any questions? Anything else I can help you guys with to prepare? Again, remember, the exam is available as early as 7 on Wednesday. You do not have to start. You can do them in any order that you want. Take a break in between them. But make sure you are giving yourself enough time to be able to complete these. This is, I honestly don't know how hard this exam is going to be. I make it in a way similar to the way I make the other exams, but essentially what I do when I make your exams is I select the questions that I want to ask. And some of that is based on randomness because, again, I like some questions better than others. Some of it emphasizes things that were emphasized better in this class, or maybe when I described something, I thought it was particularly good. And so that's the essay question that I decide to use. That's not going to be the case in this one. And for this exam, every single essay question I could think of for this material was put in a bank, right? And there's different sections. So like I said, you know you're going to have a pathway question. So maybe there's only 4 options for that one. But there's plenty of other questions and other processes that are going to be there. I probably wrote close to 50 essay questions for this between the 2 sections. And you're getting 8 of them. And which 8 you get are completely random. All right, there are 100 multiple choice questions, of which you're getting 20. And again, so the lab exam, there's almost 200 pictures on this lab exam. God, I'm doing my muscular system in the other class. Every origin, every insertion, every action, I have over 300 lab questions on their lab exam that they're taking for theirs. So it is definitely a unique experience. I have no idea what this is going to turn out like. So I'm very interested to see how this works. We'll see how it goes from there. Again, I tried to set it up so that it is equivalent to our other exams, but when we look at the end results of this, we'll see if things need to be adjusted. So we'll go with that. It is, you've all taken the practice exam, so hopefully, or many of you have taken the practice exam. I know not everybody got a chance to, but I will be available all day. I'll be checking my email. So if you have any problems or anything, or you're having difficulty getting on, hopefully that practice exam helped us to get rid of most of those issues. But I wish you luck. And I'm also curious to see how many people show up, because obviously we are still enrolled at 29. But as you look around, there's only 14. I think at most we had 16 at some point today. So I'm curious to see how many people are still around to take this. And so, yes, so and to answer the question, yes, everyone's exam is going to be different. everybody's exam is going to be different. Everybody's exam is going to have different questions. Everything is different. So yes, everybody's exam. Hopefully there is enough randomness to it that nobody's going to get all the hard questions, right? But theoretically, is it possible? Yes. But because everybody's test is completely random and how it's going to be assigned. Well, not completely. I've got blocks of questions, but everybody gets a completely random. Nobody's exams are going to be identical. And even like, for instance, the multiple choice, even if you get 3 multiple choice questions that are the same as somebody else's, yours may be 1, 2, and 5. Somebody else's may be 24, 28 and 30. So their order where they're located is going to be different as well. Yes, I'm happy to answer any questions to help you to be successful for the exam, and I'll continue the recording for that as well. So this is the rest of the new material, but I'm happy to answer any questions that you guys have on anything that I can help you to be successful for the exam, either on the process or on the content. All righty. All right, so let me have it. What are your questions?
I did have a question, I guess. If urine is sterile, how come when they say you can get an infection if you hold your pee too long? I know that it can, if it's in your urethra because you're holding it too long. I mean, is that accurate or?
Yeah, exactly. So again, think of it this way. When it is being released from the collecting duct into the minor calyx, the urine is sterile. But you're right. It then travels through the ureter to the bladder and is stored in the bladder. And the bladder is open to the outside world, and is being open to the outside world, bacteria can get in. Bacteria is in your urethra. For instance, if you give a urine sample, they typically don't want the first urine that comes out. They want you to expel some of the urine and then stop your flow and then fill the cup. And the reason they do that is that the first urine you produce is going to, clean the urethra and get the bacteria out there, so that what you're getting is that sterile or more sterile sample from what's been in your bladder as opposed to what's in your urethra. So yes, so the longer the urine is in your bladder, the more opportunity it has to interact with bacteria from the outside world, and the more likely that you could get an infection as a result of that.
Oh, okay. Thank you.
Yep. I can try to do a histology review. I don't have the best resources for that. Let me see what we've got. What I would say, the 2 resources that are probably the best for this are the anatomy lab, the PAL thing on your. So here, let's do this. So the PAL system on the anatomy lab that we have, that professional or whatever P stands for, anatomy lab on your mastering A&P. But the other resource that we have that can be good for histology would be probably that CRC site, the virtual anatomy lab. So if I remember correctly, there's always some histology on this. I don't remember if we looked at it. If we look at it here for the respiratory system. Okay, so again, that's the esophagus that we see up here. Here we see a little bit of the hyaline cartilage. So this would be the trachea. And again, the nice thing about this is they have the pictures, but then they also then show us the labels for this as well. This is indeed the trachealis muscle. Remember, the trachealis muscle is a little bit of smooth muscle. Remember, our cartilage in our trachea is C-shaped, and there's a little bit of smooth muscle that connects the 2 ends of it that helps to contract and dilate the diameter of the trachea. So yes, the fact that this is the hyaline cartilage and then we can see the smooth muscle coming off of it, it kind of is an obvious example, but I like the full view where you can see them together to really see the trachealis muscle. And again, I haven't looked at this, so I don't know what is here that we would have to look at. Here's a closer view. We see that hyaline cartilage, a little bit of the perichondrium around it, the submucosa. Here we can get a nicer view of that ciliated pseudostratified columnar epithelial tissue. We can actually see the cilia on the surface of that. So here we see, oh, there you go. That's all the stuff that we see on that. Oh, and a nice up close view. The cilia are beautiful on that. We see that. I'm not seeing any goblet cells, but remember we can sometimes see a goblet cell in here. And again, here is that lamina propria. That would be the mucosa here and then the submucosa, but this would be the lamina propria right here, that areolar connective tissue, that this collectively is our mucous membrane. Yeah, so they don't have a good picture of the trachealis other than this one, but it can be more of a one. I don't have my chat up. Where'd the chat go? Come on, where's my chat? All right, hold on. Oh, there it is. Yeah, we can do that. Let's finish the histology first, then we can do the oxygen hemoglobin dissociation curve. So we can do that too. So let's finish the histology first. So these are not bad. The one thing I would say we're missing from this, and even in the low magnification, we can't see the goblet cell and a better view of the trachealis, but that's not bad. Here we have so alveolar sacs, that's not bad, and some individual alveoli. Ooh, nice big bronchi that we see coming in here. When I'm not seeing, all right, so yeah, so this is indeed a bronchi. We can tell that because it's got a thicker cuboidal wall to it. It also has a muscular wall to it that the alveolar sacs don't. The alveolar ducts, remember, are going to be more open spaces. And then notice this is not a respiratory bronchiole. If this was a respiratory bronchiole, remember that this would have an alveolus coming off of it, and that would allow it to participate in gas exchange. So we don't necessarily know if this is a terminal. We know it's a bronchiole because we don't see cartilage. So without the hyaline cartilage, it's just smooth muscle and a cubular wall. We know it's a bronchiole, but we don't know if it's just a bronchiole or actually a terminal bronchiole. But we do know it is not a respiratory bronchiole. So respiratory bronchiole, excuse me, would have an alveolus on it. And it looks like that is it for the respiratory system. So there's a couple things missing from that, but that's not bad. Come on. There we go. For the urinary, good renal corpuscle. We can see the glomerular space, right? Bowman's capsule or glomerular capsule, right? Made up of that simple squamous epithelial tissue, really the parietal layer of Bowman's capsule, because remember the visceral are the podocytes. We can see some proximal convoluted tubules and distal convoluted tubules. Notice this one is more clear in the lumen, whereas this one's more cloudy. So again, they're cloudy because of the villi. If I remember correctly, I think this illustration is emphasizing this as a macula densa. Yeah, but remember I posted a picture on Canvas that was a much better example of the macula densa from that as well, but we can see some of those structures there. More tubules, and again, how cloudy they are tells us what they are, although notice this is more in the medulla, which you're not responsible for, so we don't have to worry about that. Ooh, here's a good ureter. Notice just a single layer of muscle that we have there. Here we have that transitional epithelial tissue. It's at a pretty high magnification. We can see the individual cells. So that tells us this is a ureter. And it probably says it at the bottom, doesn't it? Yep. See, that was covered for me. So it shows some of those conditions here. All right. Here we have our bladder. Notice we have multiple layers of the muscle. We have those rugae, those folds, the transitional epithelial tissue. And notice we can't get the whole thing into the field of view with this, whereas, come on. Why won't you change? Oh, apparently that's it. You don't have a urethra? Okay. I wonder if they'll have a good urethra in the reproductive. Let's cheat and look there. No, what? No, of course not. It's going to be there. Male. Just kidding. The male's going to have more anatomy than you're going to be responsible for, but let's at least see if it's here. Yeah, so again, it's not a good view of this, but notice as we talked about many invaginations to it, much more glands within the lamina propria of it. But again, this isn't a good view for this. Again, you can see the big invaginations, much lower magnification. Yeah, okay. So no, so none of those are great. But like I said, there might be better in your on your anatomy lab thing on the master name P. All right, so I think that's most of the histology. It's not the best, but it's some of it at least anyway. Let me go back here and go back here. And we can answer the next question, which was about the oxygen hemoglobin dissociation curve. I think the important thing to remember is that obviously you're not going to be drawing it, but you're almost certainly going to be describing it in all likelihood. So the important thing is to remember what it is. Basically what it is, it's the relationship with basically how much oxygen can bind to a hemoglobin. All right, that's essentially what it is. It is the relationship of that. And basically there are two factors involved. The first is obviously how many total oxygen a hemoglobin can hold. And of course, what is the answer to that? What's the maximum amount that it can hold?
4.
There you go, 4, absolutely. So the most it can hold is 4. Whether it holds 4 or not is basically to be determined by the primary factor, and that primary factor is the partial pressure of oxygen in the environment. All right? So, for example, in the lungs, right, the partial pressure of oxygen in the lungs is something close to, well, we know exactly what it is, 104 millimeters of mercury. And so when the partial pressure of it is that high, then essentially it is 100% binding rate. And so obviously 4 oxygen would be bound to the hemoglobin. However, as we talked about in resting tissue, right, in that resting tissue, remember, and again, we know the partial pressure of oxygen in that environment because we need to know all these partial pressures. In resting tissue, it is about 40 millimeters of mercury. And at 40 millimeters of mercury, anybody remember what the binding rate was? Well, 75, absolutely. 75%, right? Because basically what happens is when the blood gets to that resting tissue, basically it releases 1 oxygen. However, if it was like active tissue, then the partial pressure of oxygen, like we said, could be as low as 17 millimeters of mercury. And in that case, the binding rate would be closer to 25%. Right. So that means when the blood got to that oxygen-rich blood, got to that exercising tissue, that active tissue, it would give off 3 oxygen. Whereas other blood going to an area that's not as active is only going to give off 1. The beauty of this system is that how much oxygen is released to the tissue is dependent on how much oxygen is there. The more oxygen there, the more binds to the hemoglobin, the less that is released. The less oxygen that is there, the more oxygen it is going to release, and the less it's going to hold on to. And the other thing to remember about it is it's not linear, right? Because the binding of one oxygen enhances the binding of a second, right? And that's why we get that really, and again, I know you won't be able to do this on the exam, but that's why you get that nice curve shape to that oxygen hemoglobin dissociation curve, because the more it binds to it, the easier it is for another oxygen to bind. So that's the other thing, is that it's not linear. All right? That is our oxygen hemoglobin dissociation curve. And as we talked about, this is the primary factor that determines how much oxygen binds to the hemoglobin, is how much oxygen is present. But the other part of our oxygen hemoglobin dissociation curve is that other factors can influence the curve, can move the curve. All right? And basically those other factors are all indicators of metabolic activity. All right? One can be a pH change, CO2 levels, temperature, and metabolic wastes. All of these things are other factors besides oxygen that can shift that curve one way or another, make it more likely to give oxygen or less likely to give oxygen. And the short version of that is, if some other indicator indicates that the tissue is more active, then we want to give more oxygen. And the way we show that is basically by shifting the curve, right? And what we do is we shift the curve down. So when that happens, we want to give more oxygen. So our curve shifts down. So now, even though the partial pressure of oxygen may be 40 and normally I only give 1, I want to shift the curve down so that now I'm giving 2. And remember, if we shift the whole curve down, then when we redraw it, it looks like it shifted to the right. So they call it a right shift, but technically it is curving down. All right? Conversely, if one of these other factors indicates that the tissue is less active, right, then we want to give less oxygen. Oops. And so our curve shifts up. And again, when it all shifts up, it looks like a left shift. So there you go. We explained the primary factor of it. We explained how other factors influence it, and with almost no drawing. One little sneaky curve that I threw in there. But other than that, we were able to describe it and emphasize it. And obviously you could give specific examples of this and all that as well. But at its core, those are really the two factors, the two main major concepts associated with that oxygen hemoglobin dissociation curve. What primary sets it, the primary factor that sets the curve that indicates how much oxygen is going to bind to hemoglobin or how much oxygen it's giving off, and then how we can modify it. All righty. Any more questions?
Yeah, with internal and external gas exchange, how are you gonna word the question? I guess like, or is it just to describe one or the other or both in
Sure.
relation to each other, or?
Yeah, absolutely. I mean, those are all good questions. You know, obviously, as we talked about, and again, remember the important thing when you're describing these is you can do it two ways. If you think about it, it's kind of like our joints. We talked about our joints. You could say that this is a flex at the elbow, or you could say this is a flex of the forearm.
Right.
Right? You could say the joint or the part of the body that moved. It's the same thing here. When you're talking about external gas exchange, you can talk about the two structures where the gas exchange is taking place between the alveolus and that pulmonary capillary. Or you could talk the substances where it's taking place between the air and the blood. All right. Internal, it's the tissue and the systemic capillary, or interstitial fluid and blood. So again, you can do it either way. Technically, I guess you could say alveolus and blood, although that's really, you know, not matchy-matchy. It's probably better to be matchy-matchy, because the problem in that case is when you're saying blood, you're not really specifying that it's a pulmonary capillary versus a systemic capillary. And as we know, the gases move in different directions there. But I guess you could say air and pulmonary capillary. Again, I'd probably frown at that, but I probably wouldn't mark you off for that. But it's better to be structure to structure, substance to substance when describing it. But other than that, yeah, I think that those are all good, valid questions that could be asked.
Okay.
Well, the last few people who have questions, either type them into the chat or work up the gumption to say it. I will remind you that again, I will probably get the test posted tomorrow or tonight or tomorrow. But again, you won't be able to access it till Wednesday. You can access it as early as 7. And more importantly, I will be constantly checking my email between now and the exam. So if you have any questions or any concerns, then please reach out and let me know, and I'd be happy to answer any other questions that you have in that form. All right. And again, I can either respond to you by email, or the other thing I can do is we can meet on Zoom and talk and discuss these things this way as well. I received many emails over spring break, but nobody actually came to my office hours. In fact, ironically, I had several students who actually emailed me during my office hours, so were more comfortable corresponding with me that way than coming online. But I'm also available by Zoom for office hours if you need it as well. All righty. All right. Well, I guess that is it. I will leave you to your studying then. Have a good day. Take care. I'm going to stop the recording, but I will stick around if you have any last minute questions or anything that anybody wants to ask. You'll have that opportunity. Otherwise, oh wait, hold on. I have a question coming. Go ahead. Oh, okay. I didn't see the question. I'm sorry. Can we review intrapulmonary and intrapleural pressures? Yeah, sure. There you go. There's, of course, our amazing nasal cavity, right? Pharynx, larynx, bronchi, down to my alveolus. And there's my thoracic cavity, right? That works. All right, so intrapulmonary pressure, and let's just do it out here so it's easier. Which we abbreviate P, IP. And then our intrapleural which we'll call pressure PLE, something like that. I don't know. I don't remember what the actual abbreviations are, but it'll work. Here, let's just do it this. EU. There you go. Our intrapleural pressure of pulmonary. I just did it that way. Whoops. Our intrapulmonary pressure is the one that is open to the outside world. It is the pressure that is inside of the lungs. Right? We have out here our atmospheric pressure. And that atmospheric pressure is always going to drive what happens with our intrapleural pressure. Intrapulmonary, intrapleural, is basically the pressure within the pleural cavity. Right? That is the space between the visceral pleura and the parietal pleura. This one is not open to the outside world. All right? The intrapleural is open to the outside world. So intrapleural is being driven by atmospheric pressure. Right? As this pressure changes, and again, the reason this pressure changes is because of their relationships to each other as well. But as the pressure in here changes, we are going to get the movement of air either into or out of the lungs because of the changes between the atmospheric pressure and the intrapleural pressure. Right? The other thing that we need to remember is that our intrapleural must always be less than the intrapulmonary. Right? And again, it means this is always going to be a negative pressure. And negative means less than atmosphere. But it also has to always be less than the intrapleural. The reason for that is when it's less, that means with that negative pressure on the inside, the lungs are going to expand to try to fill that space in the thoracic cavity. And so that's why when we use muscles to contract and change the shape of the thoracic cavity, the lungs go with it. So when that happens, of course, as we've talked about, our pressure, our volume in here is going to increase. And that means that the pressure is going to go down. And when the pressure goes down, we now have a gradient between atmosphere and intrapleural. And so, of course, in that case, air is going to come inside. When we relax those muscles, the opposite happens. Our lungs collapse down, or our rib cage, I should say, collapse down. As the ribs collapse down, that causes a recoil of the lungs. And as that lungs recoil, that causes the volume in here to go down. And when the volume goes down, the pressure is going to go up. And when that occurs, our air is going to move out. So basically, if you remember, what happens is our intrapulmonary pressure is sometimes going to be positive and sometimes negative. As the volume goes down and the pressure goes up, right, and the air gets forced out. As the volume goes up, the pressure goes down and air comes in. But because this is open to the outside world, while it sometimes goes positive and sometimes goes negative, air is going to move. And because of that, it will always equalize to atmospheric pressure. That's why you can't breathe in forever. You keep breathing in, but eventually what happens is you stop breathing in. And the reason you stop breathing in is because I've expanded my chest as much as it can. I've dropped the pressure as much as I can, but now all this air comes in. And as more air enters my lungs, the pressure goes back up. And once the pressure inside my lungs and the pressure in the outside world comes equal, air stops coming in. Then I relax my lungs, pressure goes up, but now as the air goes out, pressure comes back down into their equal again. And so your inhale and exhale, we produce a positive and a negative pressure inside the lungs, but it always comes back to atmospheric, always equalizes to that. The same is not true in the intrapleural. Intrapleural also gets more negative and more positive, right? It goes down and it goes up, but as we expand and contract the thoracic cavity, but air is not moving into it, so it doesn't equalize. And in fact, we never want it to equalize with atmospheric pressure. Excuse me, we never want it to equalize with the intrapleural pressure. I'm intrapulmonary. All right. Sorry, I didn't catch that the first time you wrote it. Any others? Any other question on anything? All right. Well, then I guess that truly is it. You guys have a great day. Study hard. If you have any more questions, email them to me. Like I said, I can respond to your email, or we can meet on Zoom and discuss things if you have more that you want to discuss that way. Otherwise, I'm going to go ahead and stop the recording now, and I will see you. I won't see you. I'll see you guys next week on Monday. But like I said, I'll be around between now and then, and good luck on your exams. It should be interesting. So again, make sure you leave yourself enough time. Oh, last thing again, like I talked about, make sure you are recording your work environment. Again, do not have your phone out, do not have notes out. All of those things need to be put away. Make sure you're recording your environment. If you do not record your working environment, you can lose points and even potentially get a 0 on the exam. So make sure you do that properly. All right, guys, good luck and good luck. I look forward to seeing the test results. I'll talk to you guys next week.